Vision is the sense most animals rely on more than any other. It is how predators find prey, how prey detects danger, how creatures navigate, communicate, and survive in almost every environment on Earth. Below a certain depth in the ocean, vision becomes completely useless. No light reaches it. None. Not filtered, not dim, absent. And that depth covers the largest living space on this planet. What evolution did with that constraint is one of the stranger stories in the history of life. It did not produce helpless animals fumbling through the dark. It produced some of the most effective predators on Earth, hunters that abandoned eyes entirely and replaced them with something else, something that in some ways works better than vision ever could, something that finds prey through means that have no equivalent in anything hunting on land or in sunlit water above. The deepest fish ever filmed, recorded at more than 8,000 meters below the surface, in pressure that would destroy any unprotected human body instantly, has almost no functional eyes. It is nearly translucent, and it is still the apex predator of its environment. That is not an exception. That is the pattern. Across the deep ocean, animal after animal has shed vision and built something in its place, senses so precise, so alien to anything we experience, that researchers studying them are still working out how they function. And what those senses can do in complete darkness is genuinely difficult to accept the first time you encounter it. In tonight's journey, we go down past the point where light exists and meet the hunters that decided they did not need it, far below the reach of storms, below shipping lanes and whale song, and the final drifting shadows cast by surface waves. Sunlight begins to fail in stages so gradual that no boundary marks its disappearance. Red vanishes first, absorbed within the upper layers of seawater. Orange follows, then yellow, then green. Blue persists the longest, thinning into a faint mineral haze until even that last surviving color dissolves into black water. Under ideal conditions, sunlight reaches approximately 1,000 meters before ending completely. Beyond that depth, there is no dawn, no moonlight, no horizon, no flicker from the sun above. The bottom of the Mariana Trench has existed beneath permanent night for the entirety of Earth's history. Not a single photon of natural sunlight has ever touched it. The scale of that darkness reshapes the meaning of the ocean itself. The global ocean averages roughly 3,688 meters in depth, placing the average seafloor more than 2,600 meters beneath the point where light disappears. Most of the living space on this planet, measured by volume, exists in conditions no human nervous system evolved to comprehend naturally. Forests, deserts, rivers, cities, and clouds occupy the thin illuminated film above a much larger world where vision lost its authority long ago. Eyes are expensive organs. They require constant metabolic investment, not only in the retina itself, but in the neural tissue needed to process visual information. A study on the Mexican blind cavefish measured the oxygen demands associated with eyes and their related brain structures, and found that vision cost approximately 15% more metabolic energy than blindness. Near the surface, where food is comparatively abundant and light carries immense survival value, that cost remains worthwhile. In the deep sea, where a meal may drift past only once in days or weeks, 15% becomes the difference between persistence and starvation. Over immense spans of evolutionary time, the arithmetic remained brutally simple. Individuals born with slightly reduced visual systems consumed slightly less energy. Those individuals survived longer during famine conditions and passed those traits forward. Generation after generation, the visual apparatus diminished. Retinas shrank, optic lobes reduced, eyes clouded over, sank beneath skin, or disappeared entirely. The deep sea did not merely ignore vision, it selected against it with relentless consistency. What replaced sight was not emptiness, nor confusion, nor a lesser version of perception. Other senses expanded into the vacancy left behind. Pressure, chemistry, vibration, and electricity became the architecture through which deep-sea predators understood their environment. A movement through water carried shape. A dissolved molecule carried direction. The pulse of a distant heartbeat carried location. The ocean itself became the medium of perception. One sensory system in particular spread quietly through this dark world with extraordinary effectiveness. Running along the length of most fish bodies is a structure known as the lateral line, a network of fluid-filled canals and microscopic sensory cells capable of detecting tiny disturbances in surrounding water. In shallow water species, it functions as support, assisting balance and awareness beside vision. In the deep sea, it assumed a far greater role. As light vanished and eyes deteriorated, the lateral line expanded in importance until it became one of the primary ways an animal could locate another living thing in absolute darkness. Its existence remained largely invisible to human understanding for centuries, because humans possess no direct equivalent. The nearest comparison lies somewhere between touch and hearing, though neither description fully captures it. A fish resting motionless in black water can perceive the displacement caused by another animal swimming nearby without seeing it, hearing it, or physically touching it. The water itself carries the information across the body surface. Laboratory experiments eventually revealed how complete this transition had become. Researchers blinded predatory fish and found that the animals continued hunting successfully with little impairment. Only when the lateral line was chemically blocked did the behavior collapse entirely. Prey remained physically close, yet the fish could no longer locate it. In the deep sea, the lateral line is not an enhancement layered over vision. It is the replacement, ancient and precise, waiting silently beneath the skin of nearly every predator moving through the dark. Inside a laboratory tank, lit from above by pale fluorescent light, researchers altered the bodies of predatory fish, one sensory system at a time. The eyes were neutralized first. The fish continued hunting normally, turning toward prey with the same sudden precision they had shown before blindness. Small movements in the water still drew them forward. Distance still resolved itself correctly. Strikes still landed. Only when cobalt ions were introduced to disable the lateral line did the behavior collapse. Prey drifted nearby untouched. The fish circled aimlessly through the water, unable to orient toward movement, occurring only centimeters away. Vision had mattered far less than expected. The lateral line had mattered almost completely. Along the sides of most fish runs a canal system so delicate that it disappears beneath the skin unless examined closely. These canals are filled with fluid and lined with tiny sensory structures called neuromasts. Each neuromast contains hair cells crowned with microscopic filaments known as cilia. When water moves across the fish's body, pressure changes travel through the canals and bend those cilia by almost immeasurably small amounts. The movement opens ion channels inside the cells, converting physical displacement into electrical nerve signals. From head to tail, the fish receives a constant stream of information about turbulence, vibration, current direction, and movement in the surrounding water. The sensitivity approaches the limits of what biological tissue can measure. The lateral line can detect water displacement as small as 1/10 of a micrometer, smaller than many bacteria drifting through the same current. A shrimp swimming several body lengths away produces enough disturbance for detection. A fish turning suddenly changes the pressure field around it before its body fully moves through space. Even the fading wake left behind by an animal moments earlier can remain readable in calm water, like a temporary pathway suspended invisibly in the ocean. The brain assembles these disturbances into something that resembles spatial awareness without resembling vision at all. No image forms. There is no color, outline, or reflected light. Instead, there exists a shifting pressure portrait built continuously from motion itself. Direction emerges from differences in timing across the body surface. Speed reveals itself through intensity and rhythm. Size can be approximated from the shape of the displaced water. A nearby organism becomes not a visible object, but a moving geometry of force and turbulence. The ocean presses against the body at every moment, and hidden within those pressures is location. Near the surface, where sunlight still shapes behavior, the lateral line functions quietly beside the eyes. In deep water, the balance changes. The canals widen, neuromasts become larger and more numerous. Entire skull structures shift subtly to accommodate expanded sensory pathways. Evolution did not preserve the lateral line merely as compensation for blindness. It invested in it aggressively, refining it across millions of years until pressure sensing became one of the primary ways predators interpreted reality itself. The cells responsible for this ability are ancient beyond easy comprehension. The same cilia-based hair cells lining the lateral line also exist within the human inner ear, where they govern hearing and balance. When a person lying awake in darkness notices the soft movement of air through a room or hears the faint settling sounds of a distant floorboard, the underlying cellular machinery traces back toward the same evolutionary origin as the fish detecting movement through black water thousands of meters below. The structures diverged over immense spans of time, adapting toward different environments, yet they still rely on the bending of microscopic hairs, translating movement into sensation. Even so, the experience produced by the lateral line has no direct human equivalent. It is not hearing spread across the skin, nor touch projected outward into open water. It resembles a full-body awareness of surrounding space, a continuous perception of movement pressing against the animal from every direction simultaneously. The boundaries between body and environment become softer, threaded together by pressure itself. In some deep-sea predators, this sense has expanded to such refinement that the animal appears less like a hunter than an instrument waiting for a signal. Motionless for hours, suspended above the seafloor or standing against the current, it allows the ocean to move around it until the water changes in just the right way. Across certain regions of the Atlantic floor, where the sediment lies flat beneath cold currents drifting through darkness, a thin shape rises above the seafloor and remains there for such long periods that it can appear rooted in place. The body does not sway. The head points steadily into moving water. Hours may pass without visible motion. Small organisms drift through the current around it, and still the animal waits, balanced on three narrow extensions touching the mud below. It is not searching. It is listening through the water itself, holding position until some faint disturbance arrives from upstream. This animal is Bathypteros grallator, known more commonly as the tripod fish. Its scientific name carries traces of several ancient languages. Bathypteros from Greek roots meaning deep and feathered. Grallator from the Latin word for stilt walker. The name fits with unusual precision. The fish spends much of its existence elevated above the abyssal floor on structures so elongated and delicate that they resemble walking supports more than fins. The body itself reaches only around 30 centimeters in length, pale and narrow against the surrounding dark. Yet the supporting rays extending beneath it grow to nearly 90 centimeters, roughly 3 times the length of the body they hold upright. Two elongated pelvic fin rays brace the front, while a modified lower tail ray forms the third support behind, creating a stable tripod arrangement capable of anchoring the fish against slow, deep ocean currents. Standing at full extension, the animal reaches close to a meter in height, transforming a relatively small fish into a tall and motionless structure suspended above the seafloor. Its eyes remain visible, though only barely functional in the ordinary sense. The retinal tissue has degenerated to such an extent that image formation appears extremely limited, almost absent. Yet evolution did not remove the eyes completely. The remaining retinal surfaces orient forward and backward rather than outward to the sides. Researchers believe this unusual arrangement allows the fish to detect a brief point source of light directly within the narrow corridor surrounding its body. A tiny copepod releasing a flash of bioluminescence while drifting into reach may produce enough signal for the tripod fish to register its presence. It cannot survey landscapes or track distant movement. It can detect a single pulse crossing the threshold of contact. Far more important than the eyes are the pectoral fins projecting ahead of the body, like paired antennae. Held upright in the current, they contain dense concentrations of neuromasks connected to the lateral line system. Water passes first across these fins before reaching the body itself, carrying pressure information from the surrounding environment. Tiny crustaceans and drifting zooplankton entering the sensory field create disturbances that travel instantly through the canals beneath the skin. The fish responds with abrupt efficiency, striking forward without turning and without abandoning its perch. The movement lasts only moments before stillness returns completely. In the deep sea, where energy governs survival with unusual strictness, this hunting method becomes almost mathematically elegant. Pursuit requires muscle activity. Searching requires time. Constant swimming burns resources that may not be replaced for days. The tripod fish solves the problem by removing nearly all unnecessary movement from its existence. It allows the current to perform the labor of transport. Food arrives carried by the ocean itself. The predator waits. Its reproductive strategy follows the same logic of scarcity and adaptation. The tripod fish is a synchronous hermaphrodite, capable of producing both sperm and eggs simultaneously. Encounters between individuals occur infrequently across such enormous stretches of deep ocean floor, and evolution reduced the risk of missed opportunity wherever possible. Any meeting between two compatible animals can result in reproduction. Isolation no longer depends entirely on chance encounters between separate sexes. Even solitude itself became negotiable. The result is a creature shaped almost entirely by patience, a predator that survives not through speed or force, but through stillness, refined across immense spans of time. Other hunters descending through the dark followed a different path entirely, remaining active, mobile, and relentlessly searching through the ocean with senses built around chemistry rather than waiting. 300 million years ago, long before mammals appeared, and long before the first dinosaur stepped across the continents, hagfish were already moving through ancient seas with bodies recognizably close to the forms they still carry today. They survived the Permian extinction, when roughly 96% of marine species disappeared from the planet. They remained through the collapse of reef systems, the rearrangement of continents, and the repeated transformation of surface ecosystems above them. Across spans of time so large, they resist ordinary comparison. The hagfish endured with only minor alteration, continuing a style of life established before forests of flowering plants existed anywhere on Earth. Its body appears almost unfinished by the standards of most vertebrates. Long and flexible, lacking scales and lacking jaws, The hagfish moves with slow contractions that resemble neither the swimming of fish nor the crawling of eels entirely, but something older than both. Around the mouth sit four pairs of fleshy barbels rich with sensory receptors, constantly testing the surrounding water and sediment. The eyes lie buried beneath pale skin, without lens or iris, capable only of distinguishing the broad difference between light and darkness. By contrast, the olfactory regions of the brain are immense. The hagfish does not organize its world around sight. It organizes it around chemistry. For much of recorded scientific history, hagfish were regarded primarily as scavengers. They entered dead whales through natural openings or soft tissue, consuming the carcass from within while remaining protected inside the body cavity. Deep-sea footage often showed them arriving at carrion in dense numbers, twisting through decomposing flesh in slow knots of movement. The classification fit comfortably with what researchers observed, and for decades the idea remained largely unchallenged. Then, in 2011, underwater camera footage published in scientific reports revealed behavior no one had documented before in this lineage. A hagfish moved deliberately across the seafloor, lowering its barbels repeatedly into the sediment as though reading invisible traces beneath the mud. It located a live burrowing fish hidden below the surface, forced its way into the burrow, seized the animal, and consumed it. The sequence was not opportunistic feeding on the dead. It was active predation carried out almost entirely through chemical and tactile information. The process begins far from the prey itself. Dissolved amino acids leaking into the surrounding water form faint chemical gradients drifting with the current. The hagfish detects these gradients and moves upstream toward increasing concentration, adjusting direction continuously as the signal strengthens. Close to the source, the barbels sweep across the sediment, gathering both tactile texture and chemical detail simultaneously. Once the burrow entrance is identified, the hagfish inserts its body into the confined space and grips the prey using 4 rows of keratinous teeth arranged on dental plates inside the mouth. Though jawless in the conventional vertebrate sense, the hagfish generates remarkable force through specialized musculature. Deep protractor muscles drive the dental plates backward against tissue with a bite force comparable to, and in some cases exceeding, several jawed vertebrates including wrasses and turtles. The feeding motion resembles rasping more than biting, yet the mechanical power involved is substantial. A body plan older than bone continues functioning with extraordinary efficiency. Even digestion extends beyond the boundaries expected of vertebrate biology. Hagfish absorb dissolved nutrients directly through the skin lining their body wall, while simultaneously processing consumed material through the digestive tract. Burrowed deep inside carrion or prey tissue, the outer surface of the animal participates in nutrient uptake alongside the gut itself. Feeding occurs across the entire body. Recent genomic analysis of the Pacific hagfish, Eptotretus burgeri, revealed an enormous investment in chemical perception, including dozens of olfactory receptor genes and an exceptionally large repertoire of vomeronasal receptors associated with detecting molecular cues in the environment. For the hagfish, chemistry forms geography. Direction, identity, distance, and opportunity emerge through dissolved traces, drifting invisibly through cold water. Despite its soft appearance, the hagfish proves extraordinarily difficult for predators to kill. Attacks rarely proceed as expected. The body responds with a defensive reaction so rapid and so mechanically unusual that even modern engineering laboratories continue studying the material involved, searching for ways to imitate what this ancient animal has already perfected. The reaction begins before injury occurs. A predator closes its jaws around the body of a hagfish. Pressure compresses the skin for a fraction of a moment, and almost instantly the surrounding water changes state. Along the hagfish's sides sit up to 200 specialized slime pores, each capable of releasing defensive material in less than half a second. The response unfolds faster than most vertebrate reflexes. It is not triggered by pain or tissue damage, but by simple mechanical pressure, a biological tripwire responding before the bite fully closes. Inside each pore are two separate substances stored in compact form. One consists of mucins, long protein molecules that hydrate rapidly in seawater and create thick mucus. The other consists of tightly coiled protein threads packed within specialized cells like microscopic springs held under compression. Released together into surrounding water, the threads begin uncoiling immediately, extending to lengths thousands of times greater than their original size, while the mucins absorb water and expand around them. Within moments, the surrounding fluid transforms into a suspended network of fibers and hydrated gel. The expansion ratio borders on the absurd. Material compressed inside a single pore can swell to roughly 10,000 times its stored volume once exposed to seawater. A full-body discharge produces clouds dense enough to fill the gill chambers of nearby fish almost instantly. The slime itself contains remarkably little solid matter. Most of its mass is seawater captured inside the expanding network. The hagfish does not carry enormous quantities of material within its body. It carries instructions for reorganizing the surrounding ocean. For gill-breathing predators, the effect becomes catastrophic with extraordinary speed. Fish extract oxygen by maintaining continuous water flow across delicate respiratory surfaces. Hagfish slime interferes with that flow completely. Fibers and mucus clog the gill structures and trap water into a dense suspension that cannot circulate properly. The attacking animal begins gagging and convulsing as oxygen exchange collapses. Within 1 or 2 seconds, the predator almost always releases the hagfish and turns its attention entirely toward clearing its own respiratory system. Underwater footage captured during the 2011 scientific report study documented this sequence in living detail. A seal shark, Dalatius licha, attempted to consume a hagfish and almost immediately recoiled into violent coughing movements as slime flooded its gills. A wreckfish, Polyprion americanus, experienced the same response moments later, releasing the hagfish while thrashing through the water in visible respiratory distress. Neither predator appeared to modify its behavior afterward. The defensive system functions not through intimidation or warning, but through direct physiological interruption. The hagfish itself must then escape the material it produced. It does so using a movement unlike almost anything else in vertebrate biology. Beginning near the tail, the animal twists its body into a compact knot and gradually slides that knot forward along its length. As the knot travels, it scrapes accumulated slime away from the skin and generates leverage against surrounding surfaces. The same movement also assists feeding, anchoring the body while the mouth pulls against resistant tissue. What appears at first glance to be chaotic writhing reveals itself as a highly controlled mechanical behavior, refined over immense evolutionary time. The slime threads themselves have attracted sustained interest from engineers and materials scientists. Despite their delicacy in water, the fibers possess tensile strength comparable to nylon while remaining dramatically lighter in density. Research groups associated with military and university laboratories have spent years attempting to reproduce the material synthetically, studying both the protein chemistry and the self-assembling structure that gives the slime its unusual properties. The hagfish solved a problem of lightweight fiber engineering hundreds of millions of years before humans understood polymers at all. Everything about the system depends on chemistry, from the proteins stored beneath the skin to the seawater reactions activating them. The hagfish survives through chemical transformation and interprets the surrounding world through chemical gradients as well. In the deep ocean, dissolved molecules drift through currents carrying information continuously across the water column. Identity, injury, reproduction, movement, and death all leave molecular traces behind. Two animals evolve for reading those signals. The sea is filled with structure and direction, an immense invisible landscape written entirely in chemistry. A molecule released into seawater does not vanish immediately into emptiness. It drifts, spreads, folds through currents, and remains detectable long after its source has disappeared from view. Chemoreception is the sensing of those dissolved molecules in surrounding water, a form of perception capable of operating through darkness, through sediment, around physical barriers, and across distances stretching through entire sections of the ocean floor. Water carries chemical information differently from air. Molecules diffuse more slowly through seawater, yet they also remain organized for longer periods, held together within moving currents rather than dispersing instantly into turbulence. A chemical plume released by a damaged animal near the bottom of a trench may persist as a coherent gradient for hundreds of meters. To an organism equipped to read that gradient, the plume does not resemble random contamination. It has direction, intensity, and structure. The ocean becomes layered with invisible trails moving continuously through three-dimensional space. Different compounds carry different meanings. Amino acids leaking from torn tissue signal injury and exposed flesh. Certain lipid molecules identify particular prey species with surprising specificity. Shifts in dissolved oxygen can reveal metabolic activity nearby, tracing the presence of living bodies through subtle changes in surrounding chemistry. Reproductive pheromones drift outward from receptive individuals and spread through currents over enormous distances. The water contains overlapping channels of information simultaneously, each one carrying separate biological significance. In deep-sea predators, the anatomy responsible for processing these signals dominates the nervous system with remarkable emphasis. The hagfish brain devotes enormous proportions of its structure to olfactory processing, enlarging the regions associated with chemical interpretation while reducing investment in vision almost entirely. Grenadier fish carry dense layers of chemosensory tissue across the snout and head, sensitive enough to detect concentration differences measured in parts per billion. Their perception of the surrounding world depends less on shape and color than on gradients and signatures. The way a human city at night depends on roads and streetlights to organize movement. Laboratory studies involving Pacific hagfish revealed how precise this navigation can become. Individuals placed inside branching maze chambers moved without consistent preference when no chemical cue was present. The water remained behaviorally empty to them. Once food-related molecules entered one side of the chamber, their movement changed immediately. The hagfish turned and traveled toward increasing concentration with steady directional accuracy. When aversive compounds were introduced instead, they reversed course far more frequently and avoided the contaminated region. The animals were not wandering randomly through water. They were moving through chemical space with purpose and discrimination. Nowhere does this sensory system become more visible than at a whale fall. A whale weighing 100,000 kilograms begins altering surrounding chemistry the moment its body settles onto the seafloor. Cells rupture, tissue degrades, organic compounds drift outward into surrounding currents almost immediately. Time-lapse observations at artificial whale falls show scavengers arriving within hours, often beginning with larger and faster-moving individuals. The pattern suggests that the most sensitive chemoreceptors detect the plume at the greatest distances, locating the source while it remains far beyond physical visibility or tactile range. Long before the carcass itself is encountered, the ocean has already begun announcing its presence. For prey animals, this creates a difficult reality. Injury broadcasts location continuously. Slow death extends the signal. Every metabolic process releases trace compounds into surrounding water, and those traces drift outward, whether the animal intends it or not. A hidden body remains chemically active. Even complete stillness cannot fully interrupt the information escaping into the environment. Together with pressure sensing, chemical perception allows deep-sea predators to map movement, identity, and direction across vast distances without relying on light at all. Yet another layer of perception exists beneath both systems, one capable of locating prey that produces almost no motion and almost no chemical disturbance. Somewhere beneath the sediment, concealed entirely from ordinary detection, a living body still carries one unavoidable signature. Muscles contract, nerves fire. A heart continues sending electrical pulses through flesh and seawater alike. Every living body leaks electricity into the surrounding world. Each heartbeat, each firing nerve, each contracting muscle fiber produces weak electrical currents as a natural consequence of cellular activity. The signals are unavoidable. Life depends on electrochemical processes, and those processes radiate faint fields outward through flesh and water alike. No animal can suppress them completely. Even absolute stillness cannot erase the electrical trace of being alive. Certain predators evolved organs capable of reading those traces directly. Across the heads of sharks, rays, and several deep-sea relatives lie tiny pores known as the ampullae of Lorenzini, named after the Italian physician Stefano Lorenzini, who described them in the 17th century without understanding their purpose. Beneath each pore extends a narrow canal filled with conductive gel, leading toward clusters of specialized sensory cells. Small differences in electrical potential across the surrounding water alter the charge within the canals, triggering neural signals with astonishing precision. The animal does not merely detect movement or chemistry, it detects voltage itself. The sensitivity reaches levels difficult to picture clearly in ordinary terms. Ampullary organs can respond to electrical fields as weak as 5 billionths of a volt per centimeter. A human heartbeat, measured from roughly a meter away, produces a field thousands of times stronger than that threshold. In water, where electrical signals travel efficiently, even the smallest biological current becomes detectable under the right conditions. A hidden body buried beneath sediment continues announcing itself continuously through every involuntary contraction occurring inside it. Laboratory experiments revealed how dominant this sense becomes at close range. Researchers placed sharks inside controlled environments containing buried electrodes that mimicked the electrical field generated by a flatfish concealed beneath sand. Nearby food sources released chemical cues into the water, and visual information remained available as well. Yet when the sharks approached the area, they ignored the scent and disregarded visible surroundings, striking directly at the electrodes with remarkable consistency. The electrical field overrode competing sensory channels entirely. At the final stage of the hunt, electricity became the decisive signal. For prey animals concealed beneath the seafloor, this creates a difficult problem. A flatfish buried under sediment can suppress movement almost completely. Its outline disappears. Water currents no longer carry significant turbulence from its body. Chemical traces diminish as respiration slows. Yet the heart continues beating beneath the sand, muscles continue maintaining posture, nerves continue firing in microscopic pulses. Electroreception bypasses concealment by detecting the energy of metabolism itself. The sediment above the prey becomes irrelevant. Deep ocean species expanded this sensory system further as vision declined with depth. A study published in Scientific Reports during the middle years of the 2020s examined deep-sea chimeras, including Hydrolagus bemisi and Hariota avia, living hundreds to more than 1,000 meters below the surface. Compared with shallow water relatives, the deep-sea species possessed more extensive ampullary systems, with greater pore density spread across wider regions of the head and increased sensitivity within the sensory canals themselves. As darkness intensified, evolution invested more heavily in electrical perception. Prey species adapted in response. Some developed burst and freeze behaviors, moving rapidly for short intervals before becoming completely motionless. The strategy interrupts the continuous electrical signature associated with sustained movement and leaves predators tracking only the fading remnants of previous activity. Electroreceptive hunters adapted in turn, evolving sensitivity to those decaying patterns rather than relying solely on stable fields. The interaction became another silent arms race unfolding beneath the ocean surface, carried out through signals no human body can naturally perceive. Together, electroreception, chemoreception, and pressure sensing form an astonishingly detailed system of awareness. A predator may detect movement through displaced water, identify species through molecular traces, and confirm precise location through electrical fields, all within the same encounter. The ocean remains dark in the visual sense, yet it is saturated with overlapping layers of information continuously available to the animals equipped to interpret them. Still, one lineage of deep-sea hunters discovered a different solution entirely. Rather than abandoning light, it reshaped light into something almost no other organism could perceive, creating a private spectrum hidden inside the darkness itself. Most bioluminescent creatures in the deep ocean use light openly. A drifting jellyfish pulses blue to startle predators. Small crustaceans release glowing clouds into the water to confuse pursuit. Fish suspend luminous lures before their mouths or flash signals toward potential mates somewhere beyond visible range. Light usually functions as communication, advertisement, or deception directed outward into the surrounding dark. Yet one lineage of predators evolved a different strategy entirely. It produces light specifically so that almost nothing else can perceive it. Dragonfish, belonging to the genera Aristostomius, Pachystomius, and Malacosteus, generate deep red bioluminescence in water where nearly every other organism sees only blue. Below roughly 500 meters, red wavelengths disappear almost completely because seawater absorbs them rapidly near the surface. Blue persists deepest and dominates the visual world of the mesopelagic ocean. As a result, most deep-sea eyes evolved almost exclusively for blue sensitivity, tuned toward the wavelengths most likely to exist around them. Bioluminescent organisms followed the same pattern, producing blue light because blue travels furthest through water. Against this backdrop, red becomes effectively invisible. The dragonfish transformed that invisibility into an advantage. Alongside ordinary blue sensitive photoreceptors, Its retinal tissue incorporates a chlorophyll-derived pigment obtained indirectly through diet, likely originating from photosynthetic organisms far above the depths where the fish itself lives. The pigment shifts visual sensitivity toward red wavelengths, rarely detected by vertebrate eyes in the deep ocean. One of the strangest aspects of the system lies in its origin. A molecule associated with sunlight and plant biology at the surface becomes integrated into the sensory apparatus of a predator hunting thousands of meters below where sunlight can reach. With this adaptation in place, the dragonfish activates specialized red photophores beneath its eyes and along parts of its body, releasing faint crimson illumination into surrounding water. Small fish, copepods, and drifting krill pass through the beam without reacting because they cannot perceive it. To them, the water remains dark. The dragonfish, meanwhile, observes their movement clearly inside a hidden spectrum available only to itself and a handful of related predators. Hunting unfolds beneath a private light source carried invisibly through the ocean. Prey species had already spent millions of years evolving countermeasures against visible bioluminescent hunters. Some developed transparency so complete that little light reflected from the body at all. Others evolved ultra-black skin capable of absorbing nearly every incoming photon. Yet the dragonfish bypasses these defenses in unexpected ways. Ultra-black tissue absorbs red wavelengths as effectively as blue ones, making such animals appear as sharply defined silhouettes under red illumination. Transparency also becomes less effective when hidden structures cast subtle shadows inside an otherwise invisible body. The visual arms race continued, only now within a wavelength range most species never evolved to monitor. The dragonfish itself appears built around containment. Long and narrow, dark against the surrounding water, it carries teeth so oversized relative to the jaw that the mouth cannot close fully. The teeth project forward and outward in curved arrays, resembling the bars of a cage more than the bite of an ordinary predator. When prey enters the strike zone, escape routes narrow almost instantly. The dragonfish does not rely on crushing force. It relies on enclosure. Along the underside of the body runs another set of photophores, producing faint blue light directed downward. These organs serve an entirely different purpose. Above the fish, residual traces of downwelling light still filter weakly from distant surface waters. Predators searching upward may detect silhouettes interrupting that dim glow. By matching its own ventral illumination to the brightness overhead, the dragonfish erases its outline from below while simultaneously hunting upward through invisible red light. Two separate optical systems operate at once, one concealing the body and the other revealing prey. In the deep ocean, the dragonfish converted light itself into restricted information, a signal carried through darkness that almost no surrounding animal could intercept. Another predator lineage solved the same problem differently, abandoning personal control of bioluminescence altogether and instead cultivating living bacteria to generate light on its behalf, shaping an entire way of life around attraction in the dark. The light hanging before an anglerfish does not belong entirely to the fish itself. Inside the swollen tip of the lure, known as the esca, live colonies of bioluminescent bacteria maintained in permanent partnership with the host animal. The fish provides nutrients and shelter. The bacteria produce light. Together they form a hunting structure so integrated that the boundary between organism and symbiont becomes difficult to separate cleanly. Deep in the ocean, where energy is scarce and visibility nearly absent, an entire predatory strategy depends on farming living microorganisms inside a modified section of flesh. The lure hangs from a long dorsal spine called the elysium, extending outward above the mouth like a suspended lantern drifting in dark water. It glows softly, moving in slow patterns as the fish adjusts the spine beneath it. The anglerfish itself remains almost motionless. Its eyes are reduced and incapable of forming detailed images, yet the surrounding water still speaks through pressure changes reaching the lateral line. A small fish approaches the light. The water shifts. The signal reaches the sensory canals beneath the skin. At close range, the strike begins. The mouth opens with startling scale relative to the body surrounding it. Flexible bones and distensible connective tissue allow the jaws to expand to extraordinary angles, creating enough space to engulf prey larger than the predator itself. The stomach stretches in turn, accommodating meals approaching twice the anglerfish's body length. Long, transparent teeth curve backward into the mouth like inward-pointing glass hooks. Once the prey crosses the threshold and pressure closes the jaws, escape paths disappear almost completely. The anglerfish does not chase. It does not patrol territory or search continuously through the dark. The lure performs the work of closing distance while the predator waits. Its reproductive system follows the same logic of attraction and proximity, though carried to biological extremes unmatched among vertebrates. In several deep-sea anglerfish species, females may reach lengths exceeding a meter, broad-bodied and equipped with the full architecture of predation. Males remain tiny by comparison, sometimes measuring only a few centimeters long. More remarkably still, the male develops without a functional digestive system. It cannot sustain independent life indefinitely. From birth onward, nearly all of its biology is directed toward one task: locating a female somewhere in the immense darkness of the deep ocean. To accomplish this, the male carries disproportionately enlarged olfactory organs and nostrils relative to the rest of its body. It navigates by following species-specific pheromone trails released into surrounding currents by females. The search unfolds under severe constraints. The male cannot feed meaningfully during the process and survives only by consuming stored energy reserves while tracking the drifting chemical signal through open water. If the trail ends without success, starvation follows inevitably. When the male finally reaches a female, the encounter becomes permanent in many species. He bites into her skin and remains attached. Within days, tissues at the contact point begin fusing together. Circulatory systems merge. Blood chemistry equalizes. As the connection stabilizes, the male's independent organs gradually deteriorate. The eyes degenerate entirely. Internal digestive structures shrink away. Muscles atrophy from disuse. Eventually, little remains beyond reproductive tissue, sustained directly by nutrients flowing through the female's bloodstream. A large female may carry several males attached simultaneously, each reduced to a living extension of her body. More than 200 anglerfish species inhabit deep oceans around the world, and each lineage modifies this system differently. Some retain temporary attachment strategies. Others form permanent fusion, lasting the remainder of the animal's lives. The shape of the lure, the mechanics of the jaws, and the degree of male reduction vary across species, yet the underlying principle remains remarkably consistent. In a habitat where encounters are extraordinarily rare, evolution reshaped both hunting and reproduction around the challenge of drawing another organism close enough for contact. The anglerfish solved the darkness problem by transforming attraction itself into a survival strategy. Other predators abandoned attraction almost entirely, refining sensory systems capable of locating prey across distance without waiting for approach, including one pale hunter discovered only recently in the hadal depths of the Pacific, moving through darkness nearly 8 kilometers beneath the surface. In December of 2023, the research vessel RV Abate Molina drifted above one of the deepest fractures on Earth, while a steel lander began descending through nearly 8 kilometers of Pacific water. The expedition was organized through collaboration between the Woods Hole Oceanographic Institution and the Millennium Institute of Oceanography, targeting the Atacama Trench off the coasts of northern Chile and Peru. Attached to the lander were baited traps designed for animals already familiar to hadal researchers, the small amphipods that gather in trenches where organic material from distant waters eventually settles. Hour after hour, the vehicle continued downward through crushing pressure and permanent dark until it reached the seafloor almost 8,000 meters below the surface. The scientists expected scavengers. Hadal trenches are usually described through absence of light, absence of plant life, absence of abundant food. Amphipods at these depths were generally understood as opportunists, feeding on whatever fragments drifted slowly from the upper ocean. The traps had been prepared with this assumption in mind. When the lander returned, however, the contents carried a different story waiting inside the collection chambers. 4 pale animals rested within the traps, unlike any amphipod previously documented. Each measured close to 4 centimeters long, ghostly white beneath the lights of the recovery deck, their bodies lacking even the suggestion of pigment. No eyes existed anywhere on the head. Instead, powerful forward appendages curved outward like grasping hooks, reinforced with cutting structures, suited not for scraping dead flesh, but for seizing moving prey. Their shape carried the unmistakable proportions of active pursuit. This was not the anatomy of a patient scavenger feeding on remains delivered by gravity. It belonged to a hunter. The species would become known as Dulcibella comanchaca. The name Dulcibella marked an entirely new genus, not merely a new species within an existing group, a sign that evolutionary history at these depths still contains major branches unknown to science. Camanchaca comes from the languages of Andean peoples and translates roughly as darkness or deep mist, an acknowledgement of the environment from which the animal emerged. Lead author Johanna Weston described it as a fast-swimming predator named after the darkness of the deep ocean where it hunts. The trench itself stretches approximately 5,900 kilometers along the South American margin and descends beyond 8,000 meters at its deepest points. Dulcibella comanchaca was collected at 7,902 meters, where the surrounding pressure reaches roughly 790 atmospheres. Every square centimeter of the animal's body exists beneath weight sufficient to crush most surface organisms almost instantly. Yet within those conditions, evolution produced a predator specialized not for endurance alone, but for active predation. That detail changed more than classification. Until recently, the hadal ecosystem was often imagined as slow, sparse, and largely dependent on scavenging. Large predators, pursuing live prey, were assumed to be uncommon at such depths because energy arrives so irregularly from above. Dulcibella comanchaca challenged that assumption directly. Its raptorial appendages are structured for grabbing struggling animals. Its cutting mouthparts are adapted for processing fresh tissue. The body proportions suggest rapid movement through open water rather than passive waiting on sediment. Without eyes, the animal likely interprets its surroundings through the same invisible streams, already shaping so much of deep-sea life. Smaller amphipods moving nearby disturb surrounding water and release faint chemical traces into the current. Mechanoreception detects displacement. Chemoreception follows dissolved molecules through darkness that no sunlight has touched in Earth's history. Co-lead author Javier Solanas Gonzalez noted that although the species lacks eyes, it probably relies on other senses to locate food in the pitch-dark ocean floor environment. Dulcibella comanchaca entered scientific literature only 2 years ago. Its discovery, inside a trench, studied repeatedly for decades, did not shock deep-sea biologists as much as it confirmed an older suspicion that the darkness below remains densely inhabited by lives and strategies still unrecorded, organisms already perfectly adapted to conditions that surface animals could not survive for even seconds. Conditions solved patiently within deep-sea cells over millions of years. Long before any trench animal learned to hunt in darkness, the ocean itself imposed a quieter law, one measured not in teeth or speed but in weight. Pressure increases by 1 atmosphere, approximately 14.7 pounds per square inch, for every 10 meters of descent. At 1,000 meters, the surrounding force reaches 100 atmospheres. At 4,000 meters, it rises to 400. At 8,000 meters, it approaches 800. At Challenger Deep in the Mariana Trench, descending beyond 10,900 meters, the pressure exceeds 1,000 atmospheres, more than 1,000 times the force pressing against a body standing at sea level. For surface organisms, that weight becomes lethal long before the deepest trenches are reached. Proteins, the folded molecular structures responsible for almost every biochemical task inside a living cell, begin to deform under sustained pressures above roughly 400 to 500 atmospheres. Enzymes lose the precise shapes required for metabolism. Cell membranes stiffen and lose flexibility. Ion channels embedded within nerve cells fail to open and close correctly, interrupting the electrical signaling that allows muscles, sensory systems, and brains to function. Under sufficient pressure, life does not explode or collapse dramatically. It simply stops operating correctly at the molecular scale. Deep-sea animals survived this by altering their chemistry rather than resisting the pressure physically. Across fish, crustaceans, and other hadal organisms, researchers repeatedly find elevated concentrations of a compound called trimethylamine oxide, abbreviated as TMAO. It is a small organic molecule that stabilizes proteins and cell membranes by occupying the microscopic spaces pressure would otherwise compress. The deeper an animal lives, the more TMAO accumulates within its tissues. A fish collected at 4,000 meters contains measurably more than a related species from 1,000 meters. Species living near 8,000 meters possess some of the highest concentrations ever measured in vertebrate biology. The molecule does more than preserve the structural integrity of muscle or skin. It protects the sensory machinery already shaping the deep ocean world. The hair cells embedded inside the lateral line rely on delicate protein structures to convert water displacement into nerve signals. Olfactory receptor cells depend on molecular binding sites, precise enough to recognize dissolved compounds drifting through seawater. Electroreceptive organs respond to minute electrical differences across specialized membranes. Without stabilization, these systems would fail under hadal pressure long before the animal itself died. TMAO allows a predator not only to survive the trench, but to continue sensing within it. The same molecule preserves the body and the invisible instruments guiding it through darkness. At the surface, Humans encounter traces of this adaptation indirectly. The sharp ammonia-like odor associated with hagfish, grenadiers, and other deep-sea fish comes largely from TMAO and the compounds formed as it breaks down after death. The smell drifting from deep-sea flesh is the scent of pressure chemistry itself, a molecular signature of proteins being held together beneath hundreds of atmospheres. Evolution has altered anatomy alongside chemistry. A study published in Nature, Ecology, and Evolution during 2019 examined the genome of the Mariana snailfish and identified a frameshift mutation affecting the gene responsible for bone glue protein. The mutation interrupts normal cartilage mineralization early in development, leaving the skeleton thin, flexible, and only partially calcified. Dense bone, advantageous near the surface, becomes costly in the trench. Greater mineral content increases body weight while also amplifying compression forces. The snailfish skeleton is reduced to what is strictly necessary, enough structure to function, no more. Even these solutions appear to approach a final boundary. Biochemists estimate that somewhere between 8,200 and 8,400 meters, the concentration of TMAO required to stabilize proteins becomes toxic to cellular processes themselves. The chemistry that preserves life begins interfering with life at the same time. In August of 2022, researchers filmed an unidentified pseudoliparous snailfish at 8,336 meters, within meters of this predicted biochemical ceiling. It may represent the deepest point at which a vertebrate body can continue functioning at all. That record holder was not enormous, armored, or visually imposing. It was small, translucent, and gelatinous enough to rest easily inside a closed human fist. Yet within its environment, beneath pressures capable of deforming biology itself, the snailfish occupies the position of apex predator, surviving not through force in the familiar sense, but through extraordinary refinement of chemistry, structure, and sensation. If most surface minds were asked to imagine the supreme predator of the deepest ocean on Earth, the image would probably involve armor, size, speed, or enormous teeth emerging from darkness. It would not resemble a pale, gelatinous fish scarcely longer than a human hand, with loose skin, visible organs, and the proportions of a drifting tadpole. Yet at depths where pressure approaches the biochemical limits of vertebrate life, those familiar ideas of strength lose their usefulness. The animal that dominates there is soft because softness survives. The species is Pseudoliparus swire, the Mariana snailfish, formally described in 2017 after repeated observations within the Mariana Trench at depths reaching 8,178 meters. In August of 2022, a closely related unidentified Pseudoliparus was filmed in the Izu Ogasawara Trench at 8,336 meters, the deepest confirmed depth ever recorded for a fish, near the lower boundary where vertebrate chemistry can continue functioning at all. The snailfish remains not only alive, but ecologically successful. Below 7,000 meters in the Mariana Trench, the snailfish becomes abundant. Amphipods swarm across the Hadal seafloor in immense numbers, feeding on organic material sinking from distant surface waters, and the snailfish feeds on them in turn. This places it at the top of its environment's food structure, an apex predator in a world where sunlight vanished thousands of meters above, and where every movement occurs beneath hundreds of atmospheres of pressure. Nothing about the animal appears imposing at first glance, yet its anatomy reveals a body stripped almost entirely of unnecessary expense. The skull itself is partially open. Instead of forming a dense protective capsule around the brain and sensory organs, the cranial bones are spaced widely apart, leaving sensory tissue closer to direct contact with surrounding seawater. Channels carrying the lateral line system pass through these unfused regions, exposing mechanosensory structures with minimal obstruction. In shallower fish, stronger bone protects against impact and predation. In the hadal zone, dense reinforcement becomes metabolically costly and mechanically disadvantageous. The snailfish head is organized around sensation rather than defense. It also lacks a swim bladder entirely. Most shallow water fish depend on gas-filled organs to regulate buoyancy, adjusting internal pressure as they move through the water column. At hadal depths, gas cavities become nearly impossible to maintain. Compression would collapse them to a fraction of their surface volume. The snailfish instead relies on gelatinous, water-saturated tissues that compress at nearly the same rate as the surrounding seawater. Because the body and the environment deform together, the fish experiences little internal pressure differential. It does not resist the trench, it yields to it completely. Pigment has vanished as well. At 8,000 meters, there is no sunlight to absorb, reflect, or conceal against. Producing color becomes biologically pointless, another metabolic expense without reward. The snailfish body remains nearly transparent, its organs faintly visible beneath the skin. Resources once devoted to scales, dense muscle, and pigmentation have been redirected toward the chemistry required for survival and the sensory systems required for hunting. Genomic analysis published in Nature, Ecology, and Evolution during 2019 estimated that Pseudoliparus swirei diverged from a shallow water relative approximately 20 million years ago. Across that span of evolutionary time, nearly every feature useful near the surface was reduced, dismantled, or abandoned. The swim bladder disappeared, the skull loosened, pigmentation faded. TMAO concentrations increased to preserve proteins under crushing pressure. Sensory pores spread across the head. The animal became progressively more specialized for a world governed by darkness and compression rather than light and speed. The result is an organism that appears fragile only from the perspective of the surface. In its own environment, the snailfish is extraordinarily precise, chemically stabilized, pressure adapted, and perfectly configured for the hadal ecosystem surrounding it. Its ancestors entered the deep ocean carrying the architecture of ordinary coastal fish, and over millions of years, evolution removed everything that no longer served a purpose. What remained was not primitive, but refined, an animal rebuilt around conditions so extreme that sight, armor, and strength in the familiar sense gradually ceased to matter at all. In 2017, aboard the research vessel, investigator operated by Australia's Commonwealth Scientific and Industrial Research Organisation, a deep-sea trawl rose slowly from abyssal waters off the eastern Australian coast, carrying an animal that seemed unfinished at first glance. The body was unmistakably fish-like, elongated and pale against the deck lights, but the head created a strange pause among the scientists examining it. Where a face should have been, there appeared to be almost nothing at all. No visible eyes interrupted the smooth curve of the skin. No familiar expression could be located anywhere across the rounded front of the skull. Large nostrils occupied the space where eyes would normally sit, and beneath them, a small mouth pointed downward toward the seafloor. By ordinary human standards, the fish looked faceless. The species was Typhlonus nassus, commonly called the faceless cusk. It was not newly discovered. Preserved specimens had existed in museum collections since the 19th century, and the species itself had been formally described more than a century earlier. Yet it had not been recorded alive in Australian waters since 1873. Preservation jars had concealed how deeply unusual the animal truly was. In life, the smoothness of the head becomes profoundly disorienting because the absence is not damage or decay. The anatomy is complete, exactly as evolution intended it. The eyes still exist, but only barely. Beneath the skin of the head lie vestigial ocular structures, reduced remnants buried so deeply that they no longer break the surface at all. From the outside, they are invisible. The surrounding tissue simply grew over them during evolutionary time because the visual information they once provided ceased to matter in permanent darkness. Eyes became metabolically expensive, organs delivering little useful data. Natural selection did not remove them violently. It allowed them to fade. As the eyes diminished, other senses expanded into the space left behind. The nostrils grew large and prominent, positioned directly on the forward surface of the head, where vision usually dominates vertebrate anatomy. Chemoreception assumed priority. Water carrying dissolved molecules passes continuously across sensory tissues inside the nasal chambers, allowing the fish to construct its environment through chemistry rather than imagery. Pressure sensing systems distributed across the body complement this invisible map, detecting motion and displacement nearby. The face reorganized itself around the senses still functioning effectively in the abyss. In biological terms, a face is not merely decoration, it is a physical arrangement of sensory priorities. Across vertebrate evolution, skulls reshape themselves around the information most important for survival. A dog's elongated snout reflects dependence on smell. An eagle's forward-facing eyes support extraordinary visual precision. A bat carries enlarged ears and complex facial folds built around sound. The faceless cusk follows the same principle faithfully. Its face appears blank only because human perception expects eyes to dominate the front of a vertebrate head. In the deep sea, other forms of information replaced vision long ago. A related abyssal species demonstrates the same trend carried even further. Acanthonus armatus, commonly called the bony-eared assfish, inhabits similar depths and possesses one of the most reduced brains, relative to body size, known among vertebrates. Its scientific name refers to defensive spines projecting from the head, yet the visual system itself remains heavily diminished. The visual cortex is greatly reduced, while mechanosensory systems associated with the lateral line become comparatively expanded. Energy once devoted to image processing shifted elsewhere, redirected towards senses more useful in darkness and under immense pressure. This creates a quiet reversal in how surface animals interpret appearance. The faceless cusk is not incomplete. Nothing important is missing from it. Every structure that remains contributes directly to survival in its environment, while structures that no longer served a purpose gradually disappeared. What appears featureless from a human perspective is in fact highly specialized. The discomfort humans experience when looking at such a fish comes partly from recognizing that the idea of a normal face is not universal biology. It is simply the result of human sensory priorities written into bone and skin. The faceless cusk solved the abyss by abandoning everything unnecessary and refining everything that remained useful. Other deep-sea animals followed the same evolutionary logic beyond the face itself, reducing or transforming entire body plans until they became creatures surface biology would scarcely recognize. Animals eventually named after armor, darkness, trenches, and depths that once seemed beyond the reach of vertebrate life entirely. In 2020, a study published in the journal Current Biology examined a collection of deep-sea fishes whose skin absorbed so much incoming light that they appeared less like animals and more like openings cut into reality itself. 16 separate species were identified with what researchers described as ultra-black skin, surfaces reflecting less than 0.5% of the light striking them. Some absorbed approximately 99.5% of all incident light, including the blue bioluminescent flashes that dominate the deep ocean. At the time of publication, their skin rivaled or exceeded the absorption efficiency of the darkest human-engineered materials ever produced. The darkness did not come merely from pigment. Melanin, the same broad class of pigment responsible for black feathers, dark fur, and human skin tones, was present inside structures called melanosomes. Yet ordinary black coloration can be produced by melanin alone and still reflect roughly 10% of incoming light. These fish achieved reflectance values nearly 200 times lower. The difference emerged from arrangement rather than chemistry. Inside the skin, the melanosomes were packed into precise three-dimensional geometries rather than distributed randomly. Incoming photons entering the tissue encountered one melanosome after another in dense overlapping layers. A photon striking the first structure might be partially absorbed, but any remaining light was redirected deeper into the skin at angles that increased the probability of striking another melanosome almost immediately. Each collision absorbed more energy. Each deflection trapped the photon inside the architecture a little longer. Very few rays of light completed the journey back outward. Most disappeared entirely within the layered geometry before reflection could occur. The effect resembles a natural optical maze designed specifically to eliminate escape routes for light itself. The remarkable part is that evolution arrived at this solution independently from human engineering, solving the same physical problem through entirely different materials and structures. Around the same period, aerospace engineers developed vantablack for optical instruments requiring the elimination of stray reflected light. Vantablack achieved extraordinary absorption using vertically aligned carbon nanotube arrays. The deep-sea fishes reached nearly comparable performance without nanotubes, laboratories, or manufactured precision, relying instead on biological tissue organized through millions of years of selection. Among the species identified were the Pacific black dragon, Idiocanthus antrostomus, the anglerfish Melanocetus johnsonii, and the fangtooth Anoplogaster cornuta. These animals inhabit the mesopelagic and bathopelagic regions between roughly 200 and 2,000 meters, depths where sunlight has vanished and bioluminescence becomes the primary source of illumination. In that environment, visibility is not continuous. It occurs in sudden flashes, brief pulses, and moving signals emitted by living organisms. A single reflected glint can reveal a body shape, a fin edge, or the outline of a predator suspended nearby. Ultra-black skin interrupts that process completely. When bioluminescent light strikes the body of one of these fishes, almost nothing returns to the observer. The contours disappear. The animal ceases to register as a coherent object. Instead of reflecting shape, it absorbs the signal meant to reveal it. At the surface, black coloration often increases visibility under daylight because the object contrasts sharply against brighter surroundings. In the deep sea, the logic reverses. Against permanent darkness, punctuated only by isolated flashes of blue light, The blackest possible surface becomes a form of optical erasure. The animal is not camouflaged in the ordinary sense. It is functionally absent from the visual scene. This adaptation did not emerge in isolation. Once prey species evolved skin capable of absorbing nearly all bioluminescent illumination, predators relying entirely on reflected light encountered a problem severe enough to alter the direction of evolution itself. Detection systems based on pressure, chemistry, electricity, and other non-visual information became increasingly valuable because vision alone no longer guaranteed reliable perception. Ultra-black skin became part of a larger evolutionary argument unfolding silently across the water column. Some deep-sea organisms responded defensively by disappearing into darkness almost completely. Others moved in the opposite direction and turned the darkness itself into an active hunting environment, constructing entire predatory systems around it that have operated continuously for spans of time so immense that the modern surface world occupies only the most recent fraction of their history. 300 million years ago, long before the first dinosaur stepped across dry land, long before flowering plants spread across continents, before birds, before mammals, before grasslands or forests in any recognizable modern form, animals recognizably similar to living hagfish were already moving through darkness along the ocean floor. This is not an uncertain estimate inferred loosely from genetics. Fossils from the Carboniferous period preserve body plans so close to modern hagfish that the resemblance is immediately recognizable. The jawless mouth, the elongated body, the barbels surrounding the face, even the overall anatomy of the skull and feeding apparatus remained astonishingly consistent across spans of time so immense that human historical language begins to lose meaning around them. Part of the explanation lies not within the animals themselves, but within the environment they entered. The surface of Earth has never stopped changing. Continents collided and separated. Ice sheets advanced and vanished. Sea levels rose and fell by hundreds of meters. Entire atmospheres shifted chemically. Forests appeared, disappeared, and returned in altered forms. The shallow oceans became theaters of continual ecological upheaval, driven by sunlight, climate, oxygen levels, and the constant rearrangement of coastlines. Far below all of this, the abyss remained comparatively stable. The deep ocean has been cold, dark, pressurized, and chronically food limited for hundreds of millions of years. Water circulated, sediments accumulated, organic matter drifted downward from above, yet the fundamental operating conditions remained remarkably constant. A hagfish ancestor descending into those depths during the late Paleozoic era would have encountered physical rules broadly familiar to the descendants still hunting there now. Even the largest extinction events in Earth's history failed to alter the essential character of that world. Approximately 252 million years ago, the Permian extinction eliminated an estimated 96% of marine species. Entire reef systems collapsed, ocean chemistry destabilized, surface ecosystems nearly disappeared. Later, 66 million years ago, the asteroid impact marking the end of the Cretaceous period erased the non-avian dinosaurs and transformed food webs across the planet. The deep sea suffered disruptions during both catastrophes, but darkness remained darkness, pressure remained pressure. The abyss did not suddenly fill with sunlight or warmth. The core environmental logic endured, and organisms already adapted to that logic continued operating within it. Meanwhile, nearer the surface, evolution pursued a different trajectory entirely. During the Cambrian explosion, roughly 541 million years ago, increasingly sophisticated eyes appeared among marine animals. Vision transformed shallow water evolution into an escalating contest of detection and concealment. Better eyesight selected for better camouflage. Improved camouflage selected for sharper vision. Movement, coloration, body shape, armor, and behavioral complexity all intensified through an evolutionary loop driven fundamentally by light. The deep sea existed beneath this entire process. Sunlight never reached those depths. The visual arms race that reshaped shallow marine life simply did not apply there. While surface organisms evolved around color, pattern, and image recognition, abyssal organisms refined pressure sensing, chemical navigation, electrical detection, and energy conservation. They were not falling behind. They were solving different problems within different physical laws. Seen from that perspective, the strategies described throughout this journey stop appearing strange or primitive. The tripod fish remaining motionless within the current is not displaying a crude survival tactic, but one refined across tens of millions of years. The hagfish navigating through dissolved chemistry is not biologically simple. Its sensory world has been tested continuously since before the breakup of Pangea. These animals are not remnants bypassed by evolution. They are products of extraordinary evolutionary duration under unusually stable conditions. Human civilization measures time differently. If one civilization lasted 2,000 years before vanishing and another immediately replaced it, then 150,000 consecutive civilizations placed end to end would still occupy only a fraction of the hagfish lineage's existence. The comparison begins numerically and then dissolves into abstraction. The deep sea does not experience history in human proportions. Everything moving through those depths has been shaped by spans of time, difficult for the human mind to visualize clearly. Yet time alone does not explain the scale of the abyss. Space matters too, and the amount of this world, still unmapped, unseen, and biologically unknown, remains one of the most quietly overwhelming realities in all of natural history. The most important number is not the depth of the trenches or the pressure at the seafloor. It is the proportion of Earth that remains effectively unseen. Less than 20% of the ocean floor has been mapped at resolution, detailed enough to identify geological structures with confidence. A far smaller fraction has ever been directly observed through cameras, remotely operated vehicles, or human-occupied submersibles. Most of the planet beneath the water exists in scientific outline rather than in detail, a geography known more through inference than sight. The comparison becomes disorienting when placed beside worlds beyond Earth. The surface of Mars has been mapped at higher resolution than most of the ocean floor of our own planet. The Moon has been visited directly by 12 human beings. The bottom of the Mariana Trench has been reached by humans only 4 times. More people have traveled into outer space than have descended to the deepest point of the ocean. The biological implications follow naturally from that imbalance in access. Marine biologists estimate that between 91 and 95% of deep-sea species remain undescribed by science. Every serious expedition into abyssal or hadal depths returns with organisms previously unknown. In 2023, researchers retrieved Dulcibella camanchaca from the Atacama Trench, a predatory amphipod representing an entirely new genus. In 2022, cameras recorded the deepest snailfish ever observed. In 2021, researchers described new anglerfish species from Greenland waters, and identified a previously unknown black seedville anglerfish in the Gulf of Mexico. These discoveries emerged not from comprehensive exploration, but from brief encounters within a minute fraction of the available habitat. The scale of that habitat matters. The deep ocean contains the overwhelming majority of Earth's living space by volume. Mountains, trenches, abyssal plains, hydrothermal systems, and continental slopes extend across distances so immense that modern exploration technologies move through them slowly and expensively. A single expedition involving a research vessel equipped with remotely operated vehicles or deep landers can cost several $1,000,000. Observation windows are restricted by battery life, weather, communications limits, and the crushing mechanical demands imposed by depth. At hadal pressures, even small engineering failures become catastrophic instantly. As a result, exploration progresses in scattered points rather than continuous coverage. A vehicle descends into darkness, records a few hours of footage inside a narrow cone of visibility, retrieves samples, and rises again through kilometers of water. The amount of ocean directly observed during such a mission is tiny relative to the surrounding environment. Entire mountain ranges on the seafloor remain unvisited. Vast trench systems have never been surveyed visually even once. For that reason, discovery rates themselves become evidence. When new predators, new sensory adaptations, and entirely unfamiliar body plans continue appearing consistently within the small percentage already sampled, the reasonable scientific inference is not that unusual life is rare, but that the unexplored regions contain far more biological diversity than current catalogs reflect. The tripod fish, the Mariana snailfish, the hagfish, the faceless cusk, and Dulcibella comanchaca are not isolated curiosities extracted from a nearly empty environment. They are representatives from ecosystems, still mostly hidden. Even the biomass calculations overturn older assumptions about the abyss as barren space. Estimates of total carbon biomass suggest that the deep sea contains more living, organic matter than all surface ocean ecosystems combined. The darkness is not sparsely inhabited emptiness interrupted occasionally by strange animals. It is the largest, continuous, inhabited environment on Earth. The unknown portions are not unknowable in principle. They operate according to the same constraints already visible in the species that have been found. Pressure shapes anatomy. Scarcity shapes metabolism. Darkness reshapes the senses. Evolution repeatedly arrives at solutions favoring efficiency, detection, concealment, and patience over brute force. The patterns remain consistent even when the organisms themselves are unfamiliar. That consistency changes the meaning of the unknown. The unseen regions of the deep ocean are not random voids waiting to surprise science with impossibility. They are extensions of the same ancient world, already glimpsed through brief windows of observation, a world where survival depends less on strength or speed than on the ability to perceive what cannot be seen and endure conditions that surface life was never built to tolerate. At abyssal depths, reproduction begins with a problem so severe that, on the surface, it would seem almost impossible to solve. Two individuals of the same species may drift or crawl through territories separated by several square kilometers of darkness. No sunlight exists. Faces cannot be recognized because faces cannot be seen. Color displays, courtship dances, body posture, and visual ornamentation disappear into the black water before they begin. Yet mating continues successfully across millions of years and thousands of species. The deep sea did not remove communication. It redistributed it into forms that human senses rarely notice. The most widespread solution is chemical. Pheromones, dissolved molecular compounds released into the water by sexually receptive individuals, carry information through currents across long distances. These compounds communicate species identity, reproductive condition, and sex simultaneously. A drifting plume becomes a moving biological signature. The male anglerfish represents the most extreme expression of this system. Born with oversized olfactory organs and almost no future beyond reproduction, he navigates entirely through chemical space toward a female he has never seen. If he succeeds, he fuses permanently into her body. Across the broader deep ocean, less dramatic versions of the same principle dominate reproductive behavior. Water carries identity continuously. Other species solved the same problem acoustically. The family Macrouridae, commonly known as rat-tails or grenadiers, is among the most abundant groups of deep-sea fishes on Earth. Many possess specialized sonic muscles wrapped around modified swim bladders. When these muscles contract, they produce low drumming sounds capable of traveling efficiently through water over long distances. The sounds are structured rather than random, varying by species and likely by reproductive condition as well. A chemical plume may take hours to drift across a region. Sound arrives much faster. In the dark water, a rhythmic pulse moving through the pressure field can function almost like a voice, traveling invisibly between distant animals. Light itself remains useful too, even in habitats where most vision has degraded. Species retaining bioluminescent organs often employ them not only for hunting or defense, but also for recognition. Deep-sea squid and various fishes produce flashes whose timing, duration, color, and frequency form highly specific patterns. The surrounding water may contain countless isolated sparks from different organisms, but each species recognizes its own sequence within the broader field of biological light. The flashes operate less like illumination and more like coded signals exchanged across darkness. Some communication occurs through water movement alone. Certain deep-sea crustaceans and fishes deliberately generate patterned pressure pulses detectable through lateral line systems. These signals require proximity, yet within that short range they function as a kind of distant touch. Tiny mechanical disturbances pass through the water column and arrive across the surface of another animal's body as organized information. No light is necessary. No lingering chemical trail is required. The water itself becomes the signaling medium. Other species reduced the difficulty by altering the rules of reproduction entirely. The tripod fish and numerous additional deep-sea organisms evolved synchronous hermaphroditism, producing both sperm and eggs simultaneously. In sparse environments where encounters are rare, eliminating the need to locate a specific sex doubles the reproductive potential of every meeting. Any compatible individual encountered becomes a viable mate immediately. Evolution solved the statistical problem by removing half the constraints. Taken together, these strategies reveal something unexpected about the deep sea. Surface animals, especially visually dominant vertebrates like humans, compress enormous amounts of social and reproductive information into sight. Color, posture, facial expression, movement, ornamentation, and gesture all operate primarily through vision. The deep sea redistributed those functions across chemistry, pressure, sound, and bioluminescent coding simultaneously. Instead of losing communicative richness, abyssal ecosystems diversified it across multiple physical channels. From outside, these predators and scavengers can appear isolated, blind, or disconnected from their surroundings. From inside their own perceptual systems, the environment is structured, interpretable, and full of signals. Currents carry identity, vibrations carry intent, pressure fields describe nearby bodies. Electrical gradients reveal hidden life. Darkness does not erase information there. It changes the form information takes. Understanding that world fully requires more than cataloging anatomy or behavior. It demands trying to imagine perception organized around senses humans possess only faintly or not at all, an attempt that moves beyond biology alone and toward a deeper question about what any conscious creature is actually experiencing inside the private architecture of its own sensory universe. For hundreds of millions of years, in shallow seas and sunlit coastal water, predator and prey shaped one another largely through vision. Bodies evolved stripes, spots, reflective scales, false eyes, disruptive patterns, camouflage matching coral or sand, and movements calibrated to exploit the strengths and weaknesses of image-forming eyes. Detection improved, concealment improved in response, and the cycle repeated across geological time. In the deep sea, that contest never disappeared. It simply unfolded through different physical channels, in darkness so complete that sight became secondary or vanished altogether. Chemistry became one battlefield. Predators refined their ability to detect dissolved molecular traces drifting through currents, identifying prey through amino acids, lipids, and metabolic byproducts released invisibly into the surrounding water. In response, some deep-sea crustaceans evolved the ability to suppress portions of their chemical signature when stressed or threatened. Under duress, amino acid release rates can decrease, reducing the strength of the plume, spreading outward from the body. It is a form of concealment impossible to observe directly without instrumentation, a biological attempt to become chemically quieter. Yet predators, with sufficiently sensitive chemoreceptors, can detect not only the presence of chemical release, but the altered pattern created by suppression itself. The act of hiding becomes information. Selection pressures intensify on both sides simultaneously. Water movement became contested as well. Predators relying on lateral line systems detect pressure disturbances generated by swimming prey, translating tiny displacements into detailed spatial information. Some deep-sea squid species respond by altering how they move when danger is nearby. Instead of forceful jet propulsion producing broad turbulence trails, they reduce motion intensity and minimize displacement. Their bodies slip through the water with remarkable hydrodynamic restraint, attempting to avoid creating detectable signatures. Silence in the deep sea is not merely acoustic, it is mechanical. The surrounding water records movement continuously, and animals evolved to erase as much of that record as possible. Electrical signaling produced another escalation. Every heartbeat, every muscular contraction, every nerve impulse generates weak bioelectric fields. Some prey species counter electroreceptive predators through burst and freeze behavior, accelerating rapidly and then becoming completely still. The goal is not invisibility, but decay. A continuous electrical field provides a stable target. A fading field forces the predator to interpret diminishing information. Yet electroreceptive hunters evolved sensitivity not only to active signals, but to how those signals collapse over time. They track the fading residue of movement itself. A strategy effective against visual tracking fails against sensory systems operating through electricity. Because these contests occur across multiple channels simultaneously, the resulting diversity of sensory adaptations became extraordinary. Within the amphipods alone, researcher Karen Osborne documented a remarkable pattern among the subgroup Hyperiida. While roughly 10,000 amphipod species outside that subgroup share one general eye design, Hyperiids evolved 9 distinct eye types within a comparatively compact lineage. Some enlarged upward-facing regions. Others stretched visual surfaces into tubular forms or partitioned the eye into separate functional zones. The variability reflects an environment where different sensory solutions are continuously tested against one another under intense selective pressure. The important point is not merely diversity itself, but the rate and complexity of the forces producing it. Evolutionary arms races accelerate adaptation because every improvement in one organism changes the selective environment for another. In shallow ecosystems, these pressures often concentrate heavily around visual systems. In the deep sea, the contest unfolds simultaneously through chemistry, mechanics, electricity, sound, and bioluminescence signaling. Multiple independent dimensions of detection and concealment operate at once, each shaping the others continuously. For that reason, the deep ocean is not accurately described as primitive or evolutionarily slow. It is an active engine of adaptation running according to rules to surface ecosystems rarely encounter. Pressure gradients, dissolved molecules, turbulence signatures, and electrical traces become the raw material of survival. The organisms produced by those pressures inhabit perceptual worlds organized around information humans can barely sense directly. To move through such a world is to exist inside an environment structured by signals largely invisible to human awareness. Darkness, there is not emptiness, but background. Chemistry becomes geography. Pressure becomes texture. Electrical fields become traces of nearby life. The arms race shaping those perceptions has continued for spans of time longer than most surface ecosystems have existed in their present form, creating creatures whose experience of reality may differ from ours as profoundly as life underwater differs from life beneath the open sky. In 1977, the research submersible Alvin descended into the darkness of the Galapagos Rift, carrying an assumption that every branch of biology shared at the time, complex ecosystems required sunlight. Whether through forests, plankton blooms, coral reefs, or drifting marine snow produced by surface photosynthesis, all known food webs ultimately trace their energy back to the sun. Then, beneath more than 2,000 meters of water, Alvin's lights revealed towers rising from the seafloor like industrial chimneys, pouring black mineral clouds into the surrounding ocean, surrounded by dense clusters of living animals where no sunlight had ever reached. These structures were hydrothermal vents, fractures in the ocean crust where seawater seeped downward through cracks in the seafloor and encountered magma-heated rock far below. The water returned upward, superheated to temperatures exceeding 350 degrees Celsius, carrying dissolved hydrogen sulfide, methane, iron, copper, and other minerals stripped from the crust itself. When the fluid erupted back into the near-freezing deep ocean, minerals precipitated instantly, building tall chimney structures over time. Black smokers grew slowly through continuous deposition, layer by layer, in complete darkness. The remarkable part was not the geology, but the biology surrounding it. Instead of sparse scavengers drifting through empty abyssal plains, the vents supported dense communities of shrimp, worms, crabs, mollusks, fish, and octopuses. The energy sustaining them came not from sunlight, but from chemosynthesis. Specialized bacteria oxidized hydrogen, sulfide, and other dissolved compounds, using chemical reactions to produce organic carbon. These microbes form the base of an entirely self-contained food web powered by geothermal chemistry rather than solar radiation. For the first time, scientists documented a complex ecosystem running independently of sunlight at every stage of its energy chain. Around the vent outflows gathered immense swarms of the shrimp Rimicaris exoculata, sometimes packed in densities reaching thousands per square meter. Their shallow water relatives possess compound eyes suited for conventional image formation, but these shrimp evolved something entirely different. Across their backs sat flattened photoreceptive organs capable of detecting faint infrared thermal radiation emitted by the vent structures. The adaptation allowed them to navigate an invisible landscape of heat gradients. Too close to the vent core, and temperatures became lethal. Too far from the warm flow, and the bacterial growth they depended upon diminished. They occupied a narrow thermal boundary between roughly 10 and 40 degrees Celsius, guided through darkness by temperature itself. Predators moved through these structures as well. Pompeii worms, Alvanella pompejana, built protective tubes directly on vent chimney walls in temperatures approaching 80 degrees Celsius, among the hottest conditions known to support complex animal life. Carnivorous polychaete worms hunted along the vent surfaces, navigating through overlapping gradients of heat and chemistry. Nearby, vent-associated octopuses searched among mineral towers using mechanoreception and chemoreception, extending their arms through fissures and current channels where smaller animals gathered. The discovery transformed more than marine biology. Before Alvin reached the Galapagos Rift, the prevailing assumption across science held that sunlight was an unavoidable prerequisite for large-scale ecosystems. Hydrothermal vents ended that idea permanently. Life required liquid water, energy gradients, chemistry capable of sustaining metabolism, and sufficient time for evolution to work upon those conditions. Sunlight was one pathway, not the only one. That shift carried implications far beyond Earth's oceans. Beneath the ice shell of Europa and beneath the frozen crust of Enceladus, scientists already suspected the existence of deep subsurface oceans in contact with geologically active interiors. After 1977, the possibility of ecosystems existing there no longer belonged entirely to speculation. The mechanisms had an earthly example. Hydrothermal vent communities revealed that the deep sea does not merely survive without the surface world. In some places, it operates independently from it, generating its own energy, sustaining its own predators, and maintaining ecological systems governed by conditions few surface organisms could tolerate for even moments. Far below the reach of sunlight, the planet carries on another version of life entirely, one organized around heat, chemistry, pressure, and darkness, continuing steadily beneath the familiar world overhead. Long before most humans had ever seen one alive, grenadiers were already ruling the abyss in numbers so overwhelming that nearly every baited camera lowered into the deep ocean eventually found them. They emerge from the darkness slowly, heads first, pale eyes reflecting stray bioluminescent flashes, long tapering tails trailing behind like strands of smoke in cold water. Across the Atlantic, Pacific, Southern, and Indian Oceans, from depths beginning around 200 meters to trenches descending near 7,000, the family Macroridae has become one of the most consistently abundant groups of deep-sea fish ever recorded. Study after study, expedition after expedition, the result repeats with almost monotonous reliability. Lower food into the abyss, wait long enough, and rat tails arrive. Their bodies appear unfinished by surface standards. The head is large, almost oversized relative to the narrowing body behind it, which tapers gradually into the thin whip-like tail responsible for the common name. Yet nearly every visible structure is organized around sensory efficiency rather than hydrodynamic elegance. Raised ridges cross the skull in branching patterns, marking the pathways of enlarged lateral line canals beneath the skin. Water movement passes across these structures continuously, carrying minute pressure information into neuromasts distributed throughout the head. The snout is lined with dense chemosensory epithelium capable of detecting dissolved molecular traces drifting through the water column. Their eyes remain functional, though less for detailed image formation than for detecting flashes of bioluminescence against the surrounding dark. In the abyss, a single flash may represent food, competition, or danger. Precision matters more than clarity. Sound also moves through their world. Wrapped around a modified swim bladder are specialized sonic muscles capable of rapid contraction. When activated, the fish produces low drumming pulses transmitted efficiently through deep water. These sounds vary by species and appear linked to reproductive behavior and social signaling. In calm abyssal conditions, acoustic communication can travel farther than visual signals and far faster than slowly drifting chemical plumes. The deep ocean, silent to human ears, carries structured biological sound through immense distances. Because grenadiers are so numerous and so efficient at locating food, they shape the competitive architecture of the abyss itself. When a carcass sinks into deep water, chemical gradients begin spreading outward almost immediately. Grenadiers detect the plume quickly and converge in large numbers, often arriving before less abundant predators or scavengers. Their presence alters access for every species sharing that environment. Smaller scavengers feed around them. Larger predators feed among them. The timing of arrival at a food source can determine survival in a habitat where meals may remain scarce for weeks, and grenadiers dominate that timing through sheer abundance combined with highly refined sensory systems. Humans reached into this system only recently. Beginning in the 1960s, commercial deep-sea fisheries targeted several grenadier species in the North Atlantic and Southern Ocean. Marketed under names such as Hoki and Ruffy, they entered global seafood trade networks far removed from the cold darkness where they evolved. Industrial trawlers dragged heavy nets across seafloor habitats shaped over geological time, removing fish adapted to environments where growth is slow, reproduction limited, and energy conservation essential. Some grenadier species live more than 56 years. Their life histories were calibrated for stability, not extraction. Populations heavily depleted during intensive trawling in the 1970s and 1980s have shown limited recovery even decades later. In near-freezing water where food arrives unpredictably and metabolism proceeds slowly, replacement occurs on timescales. Industrial harvesting does not permit. Evolution built these fish carefully across millions of years beneath pressures and darkness almost no surface animal could tolerate. Yet, within a few human generations, entire populations declined faster than their ecosystems could compensate. The grenadier therefore exists at the intersection of two worlds. One is the ancient abyssal system that shaped its sensory biology through immense spans of evolutionary time. The other is the comparatively sudden reach of human technology into environments previously untouched by industrial activity. Increasingly, the very adaptations that allowed these fish to dominate the deep sea, their pressure sensing, chemical detection, and acoustic communication, are being studied not only as biology, but as engineering principles, hints toward ways humans might eventually navigate darkness and distance with something closer to the precision and the abyss perfected long ago. For decades, engineers studying underwater navigation kept returning to the same problem. Human technology could map the ocean through active sonar, but active sonar announces itself. A signal must be emitted, energy must be spent, and anything nearby can detect the transmission in return. Deep-sea predators solved the same navigational challenge long ago without broadcasting their presence at all. By the 1990s, naval researchers and robotics laboratories had begun studying the sensory systems of fish, not as biological curiosities, but as engineering templates refined through immense evolutionary time. The lateral line became one of the first major models. In fish, the system functions passively, detecting disturbances already moving through the water rather than generating signals of its own. Neuromass distributed along canals in the body wall register tiny pressure fluctuations, allowing the animal to sense nearby movement, current direction, wake turbulence, and the passage of other organisms even in complete darkness. Engineers studying hydrodynamics recognized that this represented a fundamentally different sensing philosophy from conventional sonar. Instead of illuminating the environment, the animal listened to the water itself. Research groups across multiple institutions began developing artificial lateral line systems, arrays of pressure sensors positioned along robotic hulls in patterns modeled after fish anatomy. These experimental systems demonstrated the ability to detect wakes left by passing objects, distinguish different hydrodynamic signatures, and track movement through turbulent water without emitting any signal. Some prototypes identified obstacles and navigated confined underwater environments entirely through passive flow detection. In effect, machines were beginning to approximate the way a deep-sea predator interprets surrounding pressure fields. Elsewhere, materials scientists focused on the hagfish. The slime threads released during defensive secretion possess an extraordinary tensile strength relative to their weight, competitive with synthetic fibers such as Kevlar, while forming spontaneously in cold seawater from compact biological precursors. Defense research agencies, including DARPA, along with university laboratories studying biomaterials, became interested in the possibility of reproducing these fibers artificially. Researchers succeeded in expressing hagfish thread proteins within bacterial cultures, yet a major obstacle remained unresolved. The proteins themselves could be produced, but recreating the precise self-assembly process by which the threads organize into functional networks proved far more difficult. The hagfish achieves, through biology alone, a manufacturing geometry human industry still struggles to imitate. The dragonfish offered another lesson. Its ability to perceive red wavelengths invisible to surrounding organisms depends on a chlorophyll-derived pigment incorporated into retinal tissue, an unusual optical adaptation among vertebrates. Engineers studying advanced sensors recognize the broader principle embedded within the system. Biological molecules evolved for one purpose could potentially be repurposed within entirely different optical contexts. Research into bio-inspired photoreceptors began exploring ways similar mechanisms might extend sensor sensitivity beyond conventional ranges, allowing detection of wavelengths previously inaccessible to standard systems. Electroreception produced perhaps the most direct technological parallel. The ampullae of Lorenzini in sharks and deep-sea relatives detect weak electrical gradients in seawater through gel-filled sensory pores connected to specialized receptor cells. Understanding how these organs process bioelectric information contributed to experimental passive electromagnetic detection technologies designed for underwater navigation and object sensing. Prototype sensors based on the same conductive gel principle achieved sensitivities approaching biological systems. Once again, evolution had already solved a problem. Engineers were only beginning to confront. Running beneath all these examples is a larger pattern. The deep sea imposed constraints of energy, scarcity, darkness, pressure, and distance upon its inhabitants for hundreds of millions of years. Every surviving sensory system represents an answer to those constraints, tested continuously across evolutionary time. Human underwater engineering faces increasingly similar conditions as exploration extends farther from sunlight and farther beneath the surface. The solutions emerging from deep-sea biology are therefore not strange accidents. They are functional blueprints produced under extreme selection pressure. Yet the most important implication moves beyond technology itself. The eyeless predators of the abyss reveal that perception is not fixed to vision, sound, or any specifically human arrangement of senses. Entire coherent worlds can be assembled from pressure gradients, dissolved chemistry, electrical traces, thermal boundaries, and hydrodynamic texture. The deep sea demonstrates that consciousness does not require seeing the world the way humans do in order to inhabit it completely. Beneath the ocean, countless animals move through darkness with extraordinary precision, constructing realities from signals the human nervous system cannot naturally perceive at all. Far beneath the seasonal turbulence of the surface ocean, below approximately 1,000 meters, the water settles into an almost permanent thermal state. Across most of the abyssal seafloor, temperatures remain between 1 and 4 degrees Celsius year after year, century after century, with almost no variation between summer and winter. Storms do not reach these depths. Sunlight does not warm them. The deep sea does not experience seasons in any human sense. It maintains one cold temperature continuously, as though the planet itself were breathing at an immeasurably slow pace. That cold reshapes biology at every level. Chemical reactions proceed more slowly at lower temperatures, including the reactions sustaining muscle contraction, digestion, nerve signaling, and cellular repair. A deep-sea fish living at 2 degrees Celsius may operate with a metabolic rate 10 to 50 times lower than a closely related shallow water species living at 20 degrees. The body consumes energy carefully and slowly, conserving rather than spending whenever possible. In surface environments, rapid movement and constant feeding often define survival. In the abyss, survival belongs to systems capable of enduring long absences rather than rapid abundance. The consequences ripple outward through behavior. A predator functioning at 1/10 the metabolic demand of a surface equivalent does not need to hunt with the same frequency. The tripod fish standing motionless in darkness for hours is not wasting time. It is operating within an energy economy calibrated to scarcity. The giant isopod, a deep-sea scavenger and occasional predator capable of reaching 76 centimeters in length, has survived documented fasting periods exceeding 5 years in captivity without feeding. Surface biology tends to interpret long fasting as crisis. In the deep sea, it is often normal physiology functioning exactly as evolved. Growth slows as well. The orange ruffy, Hoplostethus atlanticus, inhabits depths between 180 and 1,800 meters and may live for approximately 250 years. Sexual maturity arrives only around age 30. Entire human generations pass before a single fish fully enters reproductive adulthood. When industrial trawling expanded aggressively during the 1970s, populations collapsed within years because extraction operated on economic timescales while replacement operated on abyssal ones. The biology of the fish and the pace of human industry were never remotely aligned. Even movement itself reflects this altered metabolism. Remotely operated vehicle footage from the deep ocean often appears strangely slowed to surface observers. Animals pause for long intervals. Fins move deliberately. Predators remain still for minutes at a time before changing position by only a few centimeters. The behavior is frequently mistaken for lethargy or simplicity. In reality, it reflects a precise energetic calculation. In an environment where calories arrive unpredictably and movement consumes valuable reserves, stillness is efficient. Action becomes selective rather than constant. A deep-sea animal spends energy with the same caution. A desert traveler spends water. Timekeeping persists even without sunlight. Circadian rhythms evolved long before many modern ecosystems emerged, and traces of those internal clocks remain present in numerous deep-sea species despite the permanent darkness surrounding them. Without sunrise or sunset, the rhythms anchor themselves to different physical cues. Tidal pressure oscillations pass through the deep ocean with remarkable regularity. Minute temperature fluctuations ripple through current systems. Organic material descending from seasonal plankton blooms at the surface creates cyclical changes in food availability far below. The clock remains, but the signals driving it come from pressure, chemistry, and distant biological cycles rather than light. All of this means that deep-sea predators inhabit a fundamentally different temporal world. Their hunting frequency, reproductive timing, growth rates, and responses to environmental change unfold on scales unfamiliar to surface life. Some may encounter major food events only a handful of times across entire years. Others mature over decades and survive for centuries in conditions that remain nearly unchanged throughout their lives. The abyss compresses motion while stretching duration, until existence itself seems organized around patience rather than urgency. From the surface, this world can appear empty or slow simply because human perception evolved around daylight, warmth, and rapid activity. Yet beneath the cold darkness, every pause, every still body suspended in water, every long interval between movements belongs to a biological system calibrated with extraordinary precision to conditions older and more stable than human civilization itself. Everything described so far has concerned mechanism. Pressure receptors bending under moving water, dissolved molecules drifting through currents, electrical gradients spreading invisibly from muscle and heart tissue, bioluminescent organs producing wavelengths almost no animal can detect. These are physical processes, measurable and observable from the outside. Yet beneath every mechanism sits a more difficult question, one that begins not with anatomy, but with experience itself, with what it is actually like to inhabit a body built for a world humans cannot naturally perceive. In 1974, the philosopher Thomas Nagel published an essay titled, "What is it like to be a bat?" Its central idea was simple. A human being can learn every technical detail of how echolocation functions, how sound waves leave a bat's mouth, strike surfaces, and return as echoes carrying spatial information, yet still never understand what the sensation itself feels like from inside the bat's mind. Description and experience remain different things. Mechanism explains how perception operates. It does not fully reveal what perception is. That distinction settles over the deep sea with unusual force. A hagfish moving through darkness by following faint chemical gradients does not experience itself as deprived. It has never known visual space and therefore lacks any sense that something is missing. The water surrounding it contains directional information, proximity, identity, and change. Molecules released by injured tissue drift outward through currents and become orientation points in space. The animal turns toward them with precision and confidence. To the hagfish, The ocean is not an invisible world partially reconstructed through smell. The chemistry itself is the world, complete and sufficient on its own terms. The same applies to the tripod fish, suspended motionless above the seafloor. When minute pressure waves reach the elongated pectoral fins, the information arrives with extraordinary spatial detail. Direction, movement pattern, approximate distance, and behavioral signature are all present in the disturbance passing through water. The sensation is not vague or abstract. It is immediate and specific, as actionable to the fish as the sight of motion across a brightly lit room is to a human observer. The animal does not convert pressure into an imagined visual scene. It responds directly to the pressure itself, because pressure is already meaningful. From a human perspective, the abyss appears defined by darkness, because human perception is organized primarily around light. Remove vision entirely, and human orientation deteriorates quickly. In complete darkness, confidence fades from movement. Distance becomes uncertain. Even familiar spaces lose coherence after enough time without visual reference. The nervous system begins searching anxiously for information it evolved to expect, yet none of this necessarily describes the experience of deep-sea predators. The environment that disorients humans is the only environment those animals have ever known. Darkness, in that sense, may be less an objective property than a human category. To organisms dependent on sunlight and image-forming eyes, the abyss appears empty because the channel humans rely upon has vanished. But the deep ocean is saturated with other forms of information. Chemical trails drift through it continuously. Pressure fields ripple outward from moving bodies. Electrical signatures pulse invisibly from living tissue. Temperature gradients rise from vents hidden in the crust. Bioluminescent flashes pulse like coded signals through the water column. What humans interpret as absence may instead be overwhelming presence organized through unfamiliar senses. The hagfish, following a chemical plume toward buried carrion, does not pause to imagine a map. Its nervous system generates direct orientation. Movement toward the source emerges naturally from the perception itself. In the same way, a human hand reaches confidently toward a familiar doorway during a nighttime walk through a darkened home. The knowledge exists inside the body before it exists as conscious representation. These animals do not inhabit a diminished version of the human world. They inhabit different perceptual worlds entirely, structured around channels humans barely perceive and organized by sensory priorities that evolved far below sunlight. Their consciousness, whatever form it takes, is assembled not from color, shape, and reflected light, but from chemistry, pressure, electricity, and motion carried silently through cold water older than human memory. Once the question of perception enters the deep sea, the conversation changes shape entirely. The discussion is no longer only about anatomy or evolutionary adaptation, or the mechanics of pressure and chemistry moving through cold water. It becomes a question about minds, however unfamiliar those minds may be. Science cannot yet describe the inner experience of a deep-sea predator with certainty, but it can observe the complexity of the behavior surrounding that unseen interior, and the complexity itself says something important. A hagfish moving through sediment toward buried prey is not executing a single, rigid program. The animal samples chemical gradients through the water, probes surfaces with sensory barbels, adjusts direction as currents shift, evaluates openings in the sediment, enters burrows, feeds, and then reacts to predators with defensive behaviors calibrated to timing and proximity. The sequence changes according to circumstances. Obstacles alter the response. Novel conditions produce novel behavior. Somewhere inside that elongated body, sensory information from multiple channels is being integrated into a working model of immediate surroundings. The animal acts on that model continuously. Whatever else this process may be, it is not simple automation in the mechanical sense. Humans often imagine, when discussing unfamiliar animals, the same uncertainty surrounds the question of pain. In 2021, researchers writing in the journal Royal Society examined evidence concerning sentience in fish and concluded that fish possess neurological structures capable of nociception, the detection of tissue damage, along with behavioral responses extending beyond the immediate harmful event itself. Injured fish alter future behavior, avoid previously harmful environments, and show physiological stress responses, persisting after the initial stimulus has ended. None of this proves that a deep-sea fish suffers in precisely the way a human being does, yet it places the question firmly within science, rather than metaphor. The possibility cannot simply be dismissed because the environment feels alien to human intuition. Time itself appears to exist differently in these animals as well. Deep-sea species maintain circadian rhythms despite living permanently beyond sunlight. Biological clocks continue operating through cues tied to tidal pressure oscillations, subtle thermal variation, and the slow pulsing arrival of organic matter descending from surface waters. This persistence suggests a form of internal temporal organization. The animals are not suspended in an eternal present, drifting through black water without sequence or anticipation. Their bodies track duration, rhythm, and recurrence through physical signals humans rarely notice consciously in their own environment. Memory adds another layer. Studies involving electroreceptive species, including sharks, have shown that individuals can learn and retain recognition of particular electrical signatures associated with prey. Previous encounters shape later decisions. A hunting event leaves traces inside the nervous system that influence future behavior. The predator carries a history of interaction forward through time. The ocean may erase physical evidence quickly, currents dissolving trails and disturbances within moments, but the animal itself preserves something of what has already happened. Still, an immense gap remains between observing these behaviors and understanding what they feel like from the inside. No human being can directly access the perceptual world of a tripod fish balanced motionless above the seafloor, sensing minute pressure disturbances spreading through cold water. No instrument can fully translate the experience of a hagfish orienting through chemistry alone, or a deep-sea ray perceiving faint electrical pulses hidden beneath sediment. Science can measure receptors, map neural pathways, and record behavioral outcomes, yet subjective experience remains hidden behind the surface of every nervous system except one's own. This is not a limitation unique to the deep ocean. It applies to every conscious creature on Earth. The deep sea simply sharpens the problem because the sensory architectures involved differ so radically from those humans evolved around. The distance between worlds feels wider there, stretched across darkness, pressure, and senses that bypass vision almost entirely. The most honest conclusion may therefore be the simplest one. Something organized and responsive is happening within these animals, something adaptive to an environment humans can scarcely enter and cannot naturally perceive. Whether that qualifies as experience in exactly the human sense may remain permanently unresolved. Yet the question matters because it changes the meaning of the abyss itself. The deep sea becomes either a vast collection of biological machinery operating without inner life, or a hidden world inhabited by beings whose existence is subjectively real to them, even if that reality can never fully cross the distance separating their senses from ours. The human brain is built around light to a degree that is easy to forget while living beneath an open sky. Approximately 30% of the cortical surface is devoted primarily to visual processing, more neural territory than is allocated to any other sensory channel. The eyes themselves are metabolically expensive organs, consuming disproportionate energy for their size as photoreceptor cells constantly maintain electrical readiness against the flood of incoming information. Vast regions of the nervous system exist to interpret reflected photons striking the retina. Human perception, memory, orientation, language, and social recognition are all shaped by this immense biological investment in vision. What that investment purchased was extraordinary. Human eyes can distinguish roughly 10 million colors through combinations of cone photoreceptors tuned to different wavelengths. Slight angular differences between the left and right eye produce stereoscopic depth perception, precise enough to judge distance during rapid movement through complex terrain. Motion can be tracked across nearly 180 degrees of visual field. Faces become recognizable within fractions of a second. Written language, perhaps the most transformative human invention, depends almost entirely on fine visual discrimination between abstract shapes arranged in sequence. Civilization itself rests partly upon the nervous system's capacity to convert patterns of reflected light into meaning. Yet every evolutionary investment carries a cost, because energy devoted to one system cannot simultaneously be devoted elsewhere. Human beings sacrificed much in exchange for sight. In complete darkness, spatial certainty collapses rapidly. Movement slows. Orientation degrades. Humans possess little capacity to sense pressure disturbances in surrounding fluid media. Electroreception is entirely absent. Chemoreception, while functional compared with many terrestrial mammals, is weak relative to deep-sea predators that navigate three-dimensional space primarily through dissolved molecules. Even the remnants of a lateral line system survive only indirectly in the sensitivity of skin and hair to subtle air movement, a faint evolutionary echo rather than a true sensory architecture. The contrast reveals an evolutionary wager made millions of years ago. Human ancestors evolved in bright African environments where daylight was reliable, vegetation complex, and rapid visual interpretation advantageous for navigation, social interaction, hunting, and survival. Under those conditions, dedicating immense neural resources to sight produced enormous adaptive returns. The gamble succeeded so thoroughly that humans came to experience vision not as one sensory option among many, but as the foundation of reality itself. The deep sea offered no such bargain. Light never meaningfully entered most of that environment. No evolutionary path there could justify allocating large amounts of metabolic energy toward color discrimination, high-resolution image formation, or visual cortex expansion. The predators occupying those depths evolved under entirely different constraints from the beginning. Chemistry, pressure, electrical gradients, and hydrodynamic disturbance became the meaningful channels because those were the channels available. The nervous systems built there reflect that history with extraordinary precision. Below 1,000 meters, the reversal becomes difficult to ignore. The overwhelming majority of Earth's habitable volume exists beneath sunlight, extending across more than 60% of the planet's surface area and descending through kilometers of cold water where human sensory architecture performs poorly. A human entering that world without technology becomes almost helpless within moments. Vision narrows to darkness. Sound loses clear directional meaning. Chemical signals remain unreadable. Electrical fields pass unnoticed through the body. The environment ceases to feel coherent. Meanwhile, the eyeless predators described throughout this documentary continue functioning with complete effectiveness. A hagfish follows molecular trails through shifting currents. A tripod fish detects minute water disturbances through elongated sensory fins. A deep-sea shark perceives the electrical discharge of hidden prey buried beneath sediment. These are not degraded approximations of the human sensory experience. Within their own environment, they are superior solutions. The inversion carries a quiet but profound implication. Human beings tend to measure intelligence, awareness, and sensory richness against the standards of their own biology. Eyes facing forward, detailed color vision, rapid image recognition, and visual memory become unconscious benchmarks for what it means to perceive the world fully. Yet the abyss demonstrates that these traits are specialized adaptations rather than universal ideals. The sensory systems dominating the deep ocean evolved under pressures older than human existence and remain effective across spans of time and geography almost impossible to comprehend. What counts as perceptual sophistication depends entirely on the world an organism inhabits. In the deep sea, the human arrangement of senses is not advanced. It is provincial, calibrated narrowly for sunlight and open air. The abyssal predators are not missing anything essential. They are exquisitely fitted to a world that has remained cold, dark, and immense for hundreds of millions of years, and that will likely remain so long after the surface world above it has transformed into something unrecognizable to anything living today. For most of human history, the deep sea has been described as though it exists at the edge of life itself. The language surrounding it tends toward extremity: alien, hostile, abyssal, inhospitable. A realm imagined as distant from the ordinary conditions under which biology properly unfolds. Yet after moving slowly through the sensory systems, timelines, predators, and evolutionary histories of that world, another possibility begins to emerge. The deep ocean may not be the exception to life on Earth. It may be closer to the baseline from which the thin brightness of the surface briefly departs. Below 1,000 meters, the ocean covers more of Earth's surface than all continents and islands combined. By sheer volume, the deep sea forms the overwhelming majority of habitable space available to complex life on this planet. The surface world humans inhabit, with its weather systems, forests, cities, migrations, sunlight cycles, and visual ecologies, occupies only a narrow film stretched across the upper boundary of a vastly larger environment beneath it. Even temporally, the balance tilts downward. The abyssal ocean has remained cold, dark, pressurized, and broadly stable for hundreds of millions of years, while the surface repeatedly transformed through continental rearrangement, atmospheric shifts, glaciation, warming periods, asteroid impacts, and mass extinctions. The creatures adapted to that stability, endured accordingly. Hagfish persisted through biological catastrophes that erased most marine species alive at the time. Grenadiers spread across nearly every major ocean basin. Chemosensory predators occupied trenches long before mammals appeared and continued operating there while entire surface ecosystems rose and vanished overhead. The deep sea did not require reinvention after each planetary disruption because the core physical structure of its world remained intact. The darkness stayed, the pressure stayed, the cold stayed. Life there adapted not to rapid transformation, but to continuity. Seen from sufficient distance, the implications become strangely disorienting. If an outside observer arrived at Earth and attempted to identify the planet's most successful long-term animal strategies, the answer would likely look nothing like human intuition expects. Large terrestrial mammals would appear recent and geographically limited. Primates would seem extraordinarily specialized toward a narrow environmental band requiring light, oxygen-rich air, and moderate temperature stability. By contrast, the deep ocean lineages would register as ancient, widespread, and remarkably durable. Eyeless scavengers and pressure-sensing fish would not appear marginal, they would appear foundational. That reversal quietly unsettles many assumptions humans carry without noticing. The belief that sight is the dominant sense arises from a species built almost entirely around visual cognition. The belief that light is necessary for a complete world reflects the architecture of the human nervous system more than any universal biological truth. A face without visible eyes appears incomplete only because human social recognition depends overwhelmingly on eye contact and facial orientation. Slowness appears primitive, largely because human metabolism and attention evolved within environments rewarding rapid visual reaction. Even emptiness itself becomes questionable. The abyss once seemed vacant because humans lacked the senses needed to perceive the information saturating it. The deep sea answers these assumptions not philosophically but biologically. It presents living counterexamples, refined over spans of time almost beyond comprehension. A hagfish locating prey through chemistry alone. A tripod fish reading water movement with exquisite precision. A faceless cusk reorganizing its anatomy around smell because vision no longer mattered. A dragonfish constructing a private visual spectrum inaccessible to surrounding animals. None of these are theoretical arguments. They are functioning organisms that survived because their sensory worlds worked. In that sense, the abyss operates less as an alien landscape than as a mirror held beneath the surface of human certainty. It reflects back the narrowness of assumptions mistaken for universals. The human world begins to appear not central but contingent, one particular solution among countless others shaped by a temporary layer of sunlight at the top of an older planet. And beneath that layer, where pressure replaces weather and chemistry replaces color, the predators described throughout this story continue moving through darkness that is not darkness to them at all, inhabiting a world that has remained coherent, inhabited, and alive for longer than almost anything humans call familiar. Somewhere on the Atlantic abyssal plain, far below shipping lanes and storms and sunlight, a tripod fish is almost certainly standing in darkness at this moment. Its elongated fin rays are planted into soft sediment. The body angles slightly upward into a current it cannot see. Pectoral fins remain extended, like delicate sensing structures held open against the passing water. Nothing about the posture appears dramatic. There is no chase, no visible struggle, no spectacle of force. Yet this arrangement, or something extremely close to it, has persisted in the deep ocean for tens of millions of years with only minor variation. The animal waits because waiting works. The scale of that waiting becomes difficult to hold clearly in the human mind. From the earliest writing systems appearing in Mesopotamia roughly 5,000 years ago to the present day, all recorded human civilization occupies only a tiny fraction of the time hagfish lineages have already spent hunting through the abyss. Entire empires rose and disappeared during intervals that would barely register against the broader continuity of the deep ocean. Languages formed, fragmented, and vanished. Religions spread across continents. Cities expanded upward into electric light. None of it interrupted the patient sensory world beneath the water. The tripod fish itself possesses no awareness of geological history. It is not contemplating epochs while balanced above the seafloor. Its nervous system remains occupied by immediate things, by the faint pressure distortions moving through cold water, by the arrival of microscopic prey, by subtle current changes carrying information through the darkness. Yet from outside that world, looking downward across millions of years, the stillness acquires another dimension. The patience of the deep sea does not emerge from conscious reflection. It emerges from continuity. While the abyss remained broadly stable, the surface world transformed repeatedly. Dinosaurs dominated and vanished. Forests spread across continents previously barren. Ice advanced and retreated over entire hemispheres. Flowering plants reshaped terrestrial ecosystems. Mammals diversified. Primates descended from trees and began constructing symbolic cultures capable of recording their own existence. Across all of these transitions, the deep sea registered disturbance without losing its underlying character. The darkness remained intact. The cold remained intact. The pressure remained intact. Predators continued sensing movement through water older than memory. Even now, as the surface ocean warms and acidifies under human influence, the deepest layers change more slowly than almost any environment on Earth. Oxygen levels shift gradually through intermediate depths. Carbon moves downward over decades and centuries. Temperature increases spread with enormous inertia through abyssal circulation systems that take centuries to complete a single planetary cycle. The deep sea will not remain untouched forever. Nothing biological escapes planetary change entirely, yet its timescale differs profoundly from that of the surface. Long after coastlines alter and ecosystems reorganize in shallow water, something recognizably descended from today's abyssal predators will likely continue moving through darkness below. This leads naturally toward an image rather than a prediction. If one imagines the distant future of complex animal life on Earth, the final, enduring predators are unlikely to resemble the large visual hunters humans instinctively associate with dominance. The surface rewards speed, display, and rapid adaptation to fluctuating conditions. The abyss rewards efficiency, patience, and sensory systems calibrated to permanence. An eyeless, pressure-sensing, chemically guided animal inhabiting cold water kilometers beneath the surface possesses certain advantages simply because its environment changes so slowly. The deep ocean has already survived catastrophes that erased dominant surface lineages many times over. And through all of this, the tripod fish remains unaware of the beings above it. It does not know that humans lowered cameras into its world. It does not know that scientific papers described the arrangement of its fins or the mechanics of its sensory systems. It does not know that entire narratives have been assembled on the surface from fragments of footage gathered through expensive expeditions into darkness. The fish simply continues existing within the logic of its environment, balanced against the current, waiting for the next movement carried through the water. There is something quietly immense in that indifference. The deep sea does not require human recognition to remain complete. Its predators hunted successfully long before human beings appeared, and they will likely continue long after the surface world changes into forms no living person could recognize. Beneath everything temporary overhead, the dark continues with extraordinary patience, holding its ancient rhythm beneath the restless brightness above. Far below the point where sunlight dissolves into black water, the predators of the deep sea continue moving through a world that is complete to them. The hagfish follows invisible chemical gradients through sediment older than human civilization. The tripod fish balances against a current, carrying pressure signatures across the seafloor. The snailfish drifts through crushing water with a body shaped by forces no land animal has ever encountered. By every measure science can presently reach, these animals inhabit environments that are coherent, navigable, and fully real from the inside. Their world contains no daylight, no horizon, no visible landscape in the human sense. It contains chemistry, vibration, electrical disturbance, temperature variation, and the movement of water itself. It has operated under those conditions for hundreds of millions of years without interruption. From the perspective of the surface, this environment acquired a simple name: darkness. The word suggests emptiness because human beings experience darkness primarily as deprivation. When the lights fail in a room, information disappears. Orientation weakens. Confidence collapses inward. The deep sea does not experience darkness this way because its inhabitants were never built around light to begin with. Below the light line, darkness is not an absence waiting to be corrected. It is structure. It is texture. It is information carried through other channels. Predators and prey continue their ancient negotiations there without requiring illumination at all. The currents carry signals. Pressure waves shape geography. Chemistry maps the location of living things. The darkness is biologically crowded, even when it appears visually empty. Human language strains against this reality because language evolved inside a visual species. English contains thousands of words for color, brightness, shape, expression, appearance, shadow, reflection, and sight. It possesses almost nothing comparable for the direct perception of water movement through the skin or the detection of an electrical field surrounding another organism. There is no ordinary human vocabulary for building a mental map from pressure differentials alone. There is no precise word for recognizing the heartbeat of nearby prey through conductive tissue without touching it. These experiences remain outside common speech because they remain outside human biology. The deep sea contains sensory worlds that can be measured scientifically while still resisting description in ordinary language. There is, perhaps, one faint movement toward understanding available to surface creatures. Lying awake in darkness, listening carefully, the human brain begins redistributing attention away from sight. The shape of a room becomes partially audible. Distance emerges through echoes and subtle vibrations. The movement of another person nearby alters the sound texture of the air. A familiar house can be navigated with closed eyes because the body gradually assembles another kind of map beneath conscious awareness. Even this remains enormously distant from what deep-sea predators experience, because their nervous systems evolved specifically for those channels over immense spans of evolutionary time. Yet it offers the smallest glimpse of a different sensory arrangement, one in which vision no longer dominates every other source of information. And somewhere beneath the surface tonight, in water more than 4,000 meters deep, something is hunting through that arrangement now. It moves across a seafloor untouched by sunlight, through cold that has remained nearly unchanged since before human ancestors walked upright. Its world is not frightening to it. It is not isolated. It does not experience itself as trapped inside darkness. It is adapted completely to the environment surrounding it, as naturally as birds are adapted to air or forests are adapted to rain. The deep sea is not empty between scattered creatures. It is inhabited continuously by lives unfolding beyond human notice. The dark was full long before human beings arrived to describe it. It remained full while continents shifted, climates changed, and entire surface ecosystems disappeared. It will remain full after the present world overhead becomes something else entirely. There is something unexpectedly gentle in that continuity. Beneath the noise and brightness of the surface, another world has endured in enormous silence, patient and complete, carrying on without witness through stretches of time almost too large for the human mind to hold clearly. The ocean has never required human understanding in order to function. Long before research vessels crossed the Atlantic or remotely operated cameras descended through black water, the abyss was already complete. Currents moved across trench walls older than forests. Predators tracked prey through pressure and chemistry. Bioluminescent signals drifted upward through the mesopelagic dark and vanished again. The science gathered across this documentary represents decades of effort, thousands of hours at sea, pressure-resistant machinery lowered into environments capable of crushing steel, and the patient work of researchers attempting to observe a world that remained hidden for most of human history. The deep sea never resisted discovery actively. It simply continued existing at a scale and depth that made observation extraordinarily difficult. And every time those cameras arrived, every expedition found the same thing waiting below. Not emptiness, not silence in the biological sense. Life appeared consistently, adapted in forms that surface intuition would never predict, yet perfectly fitted to the conditions surrounding them. Hagfish moved through sediment by smell. Grenadiers crossed the abyss in enormous numbers, reading currents through the canals etched across their skulls. Snailfish drifted at the edge of vertebrate possibility in water heavy enough to deform proteins. Anglerfish carried living bacteria as lanterns. Amphipods hunted at depths once assumed too extreme for active predation. Entire ecosystems gathered around volcanic heat rising from beneath the crust, independent of sunlight and older than any human civilization. There is something strangely gentle in the consistency of that discovery. The deep sea did not become alive when humans reached it. It did not awaken under observation. It had always been occupied. Every trench, every abyssal plain, every hydrothermal vent field explored so far has revealed animals already living within complete sensory worlds of their own. They do not require witness. They do not orient themselves around the surface. The ocean floor does not pause when ships pass overhead. Somewhere tonight, perhaps 600 meters below a quiet stretch of Pacific water, a small fish releases a pulse of bioluminescent light into the dark. The flash lasts only a moment before the water closes around it again. To most nearby animals, the signal disappears immediately. But 30 centimeters away, suspended motionless in the water column, a dragonfish perceives it through pigments tuned to wavelengths. Almost nothing else in the ocean can detect. The red illumination reflects invisibly across the prey's body. The predator does not rush forward yet. It waits. Its nervous system is processing movement, orientation, and distance through sensory pathways refined across spans of time longer than mammals have existed. Variations of this encounter were already happening while the continent still carried unfamiliar coastlines and the first forests spread across the land. And above all of it, in quiet rooms lit only by small clocks or distant streetlights, human beings lie awake listening to stories about a world that surrounds them almost completely. The floor beneath the bed connects, ultimately, to continental shelves descending into black water where hunting never stops. The deep ocean will continue moving through its cycles tonight, exactly as it did before human language emerged to describe it. Creatures without eyes will follow chemical trails through darkness. Pressure-sensitive hunters will wait against the current. Soft-bodied predators will drift through trenches untouched by sunlight. None of them require the surface world to notice. The ocean asks nothing of the land above it. It has endured ice ages, extinctions, and rearrangements of continents without surrendering its deepest patterns. Beneath the surface noise of human history, another world continues patiently in the dark, ancient, and ongoing. The ocean keeps its secrets in the dark, and the dark is where they belong.